Good morning, everybody. My cell phone says it's 9 o'clock. So we will start the part of the second day of the seminar. It's only going to be a half day, so it'll be short and we'll have no time pressure. The audience is certainly a little bit thinner this morning. Maybe we stayed a little too late last night, but as you can see, we all had a very, very good time. So thank you to our industry partners for making it possible. So this is the 8th annual Montreal Oscillometry Summer Seminar. We have indeed confirmed the recordings worked yesterday, so everything will be posted within days on the Respiratory Effectiveness Group website. And for those of you who are relatively new to oscillation mechanics, I suggest the first 1 hour of the 2020 seminar series with a great historical overview by Charlie, wonderful modeling by Jason, and then the theory given by Zoltan. You'll never get 3 better lectures to introduce yourself to the field. So watch them, I urge you. We're still coming from the McGill University campus, not the super hospital. We moved into the room very comfortably, and these are the pictures from yesterday. And just to remind anybody who may have forgotten or who was not here yesterday, if you want to use the internet, you have to sign in as a guest on the mcgill.ca on your Wi-Fi icon, and then follow the sign-in instructions. So once again, thank you to Pharma for making this, and industry, also manufacturers, making this possible. This is again a good time last night, which I don't need to dwell on. Finally, a thank you to my 10 med students who have helped me with the logistics and will continue helping through today. The McGill University staff and the Faculty of Medicine, and of course, the Meekins Christie staff who have been taking care of me for the past 35 years. So welcome back to the seminar. We've got a great rest of the program. And I'm going to hand the podium over to Brian Ross, who's going to moderate the coming 4 talks. There's Grumpy.
Yeah, you'll put that on.
Oh, look at that. I like the t-shirt, Zoli. Not loud, but proud.
Grumpy old.
Fantastic.
So hi, everybody. Welcome back this morning. My name is Brian Ross. I'm a clinician scientist here at McGill University Health Center. I would describe myself as a first-time caller, long-time listener of this field. And I thank you to Ron Danderen for asking me to moderate, and for all of the field trips out to his lab in St. Anne's. I've learned so much. So welcome back, everybody. We're going to get started with Dr. Jason Bates from the University of Vermont, who's going to be speaking about oscillometry during mechanical ventilation of the injured lung.
Sure, take care of that. Oh yeah, me. I could take care of that. No problem.
All right.
Oh, Rob, Brian, move Zoli up to the corner just so it's maybe out of the way completely. You can just make it the silver bar on the far left there. Yeah.
Great. Well, thanks. Welcome, everybody. And thank you very much, Ron, for inviting me to this and doing this, organizing this very important conference, very important for the field. It's a pleasure again to be here. I did as instructed and wore my loudest shirt.
Well done.
Showing flamingos engaged in various immoral and possibly illegal activities. So, right. So today I'm going to talk about oscillometry during mechanical ventilation. A, to demonstrate that it can be done, and B, to show that we get some interesting data from it. And I've added the names of my co-conspirators in this particular project. We've been funded by an R01 grant over the last, about 5 years ago, and we just happily got it renewed. So we're going to be able to continue to do this. And we have a variety of expertise involved in the team, which is essential for doing this kind of work. So, right, disclosures, R01 support, usual stuff. So a little background, first of all. The medical situation we're looking at here is acute respiratory distress syndrome or acute lung injury. And when you have a patient on a ventilator with acute lung injuries, everybody knows ventilator-induced lung injury, in other words, killing them with the ventilator, is a major risk factor. And as a biomedical engineer, I see that as an engineering problem, question of managing stresses and strains in some sort of optimal way. So it suits a guy like me to think about this. The current standard of care is using so-called protective low tidal volume ventilation. So 6 milliliters per kilogram of ideal body weight, which ends up being a much smaller tidal volume than you would normally give. And the reason for that seems like a no-brainer. You're stretching the lung less than you otherwise would. You stretch it less, you're less likely to break it. Turns out, however, the situation is more complicated than that, because the mortality is still very high, depending on who you ask, 30%, some might say 40%. It's still pretty bad if you have full-blown ALS. Now, the situation or the method that we've been looking at in our group is airway pressure release ventilation. It's called ARPV, which gives sustained inspirations at fixed airway pressure, interspersed by brief expirations. I'll explain that a little bit more in just a second. But the thinking here is that if the expirations are short enough, they are protective against ventilator-induced lung injury because they don't allow enough time for epithelial surfaces to do this, so they have to be ripped apart again during inspiration. And I know from a lot of work we've done in mouse models that that is extremely damaging to the lung if it happens everywhere. In fact, I would say, in mice anyway, that is the worst thing you can do. The lung is actually pretty good. It's designed to be stretched. But repeated recruitment is very bad. So the hypothesis we're looking at here is, in fact, just what I said, that by using brief enough expirations, APRV protects against adult trauma by not giving these epithelial surfaces enough time to come into apposition, so they have to be ripped apart. We've had this theory for quite a while as to why APRV, if it's set up the right way, is protective. And in this study, we want to use oscillometry to get some direct evidence of that. So the methods were, and these experiments were done by my colleagues at the Upstate Medical University in Syracuse, where they have a sort of pig ICU and can do some very fancy experiments. They did tween lavage to 40 pigs to create an acute lung injury. They were then subsequently ventilated by this APRV mode. And the animals were divided into 4 groups, depending on whether they had high inflation pressures or moderate inflation pressures, high being 40 centimeters of water, moderate being 28, and whether they had a brief expiration or a slightly longer expiration. And by brief, I mean about half a second, just enough to get CO2 out before you start the next inspiration. And by longer, I mean not very much longer, 1 to 1.5 seconds. And so the OD minus, RD minus group, for example, is those who are protected against overdistension and not getting much recruitment and de-recruitment, whereas the plus group on the bottom were the most damaging group. And we made measurements of what I'll show you in a second, immediately after injury and every hour thereafter for 6 hours. So these were pretty major experiments that my colleagues were able to do.
Jason, not to interrupt the flow, but so what's your eye time? How often? What's the frequency of this?
Oh, the frequency, I'll tell you exactly what the frequency was. The inspiratory time was like 5 seconds.
Oh,
Something like that, 4 or 5 seconds. Yeah, 5 seconds, I would say.
yeah.
Yeah, so it's, some people describe it as CPAP with occasional releases. So APRV, the waveform looks like this. We have an extended pressure during inspiration called P high, and then a brief reduced pressure, actually 0 centimeters of water, called T low. And if you look at the flow, it does that kind of thing. So the real critical thing is how long is that T low, the time at low pressure. And the groups that we divided the pigs into had either a P high of 28 centimeters of water or 40 centimeters of water, the latter being subjected to overdistension, or a T low of about half a second or about 1 second or longer, being those who had brief enough to avoid thought, this adult trauma, and those who were long enough to sustain it. Now, we've published the basic physiology of these animals already, and what we find is that immediately after injury, I don't have a pointer. Well, anyway, you can see immediately after injury, the PF ratio, which is one of the clinical characteristics of ARDS. Prior to injury, the PF ratio was in the healthy range up there on the top left. Immediately after injury, all the groups had a PF ratio of less than 300. Oh, thank you. Yeah, there we go. Yeah, less than 300. And then the most injurious group that were getting over the standards and had repeated recruitment and de-recruitment failed to recover up to non-ARDS levels, whereas the other three groups did. So we were stratifying these animals into how well they did. So in other words, the pink groups on the bottom were getting ventilator-induced lung injury or were not being allowed to recover, whereas the other ones were. Oh, there we go. So the higher pressure group at 40 centimeters of water and longer expirations had an impaired recovery over 6 hours. So the group at Syracuse did histology, gross histology, and microscopic histology. You can see on the bottom the injured group is clearly injured compared to the less injured group and so forth. So a fairly extensive characterization of the actual injury was done. So what we did here was went back and did a more precise analogy, because the measurements that were made every hour were 10 seconds of forced oscillation being applied during mechanical ventilation. And the device that we built to do it is this. It's called a porcine oscillator. It's known as the porcellate. And so we have a mechanical ventilator in the pig, and we're measuring airway pressure and flow right at the entrance to the endotracheal tube. But in the inspiratory line of this ventilator, we had a loudspeaker encapsulated in a chamber. So there was a space behind and a space in front of the speaker. And when the speaker cone oscillates, it drives oscillations into the inspiratory line, which then go into the animal, and we measure them here. A critical practical point that we had to implement here, which is an old idea, so they didn't think of this, is that we have a relatively higher impedance connection between the front and back chambers. And the reason for that is that during inspiration, you can have a pretty high pressure in this line. And if you don't have that equilibration channel, what you do is you ram the speaker cone back up against its travel and it can't move. But by having a slow equilibration, by slow, in the order of a ventilated breath, you allow equilibration of the pressures either side of the speaker cone, so that doesn't happen. But the higher frequency oscillations of the cone, which go from 5 to 19 hertz, do go into the line and into the animal. This is a photograph of one of the early mock-ups. This was built by our biomedical engineer, Nirav Daftari, and actually he got a drum kit, like a rock drum set, and he got the snare drum casing and put a speaker in that. We eventually had to have a slightly fancier one built, but this was the early type. And you can see the oscillations come out here, and this is our mechanical one. So we tested the thing in the lab and made sure it worked.
How low are your frequencies going? Because it looks like you've got a 12-inch speaker there.
Yeah, it was a pretty big speaker. Well, this is,
Oh,
no, we can't, sorry. Anyway, we used prime numbers from 5 to 19. It's the usual kind of thing. Okay, so this is what a piece of the data looks like, and I'm showing the pressure and the flow waveforms over a mechanically ventilated breath. And you can see that we're getting pretty sizable oscillations. So those oscillations are going into the lung during both inspiration and expiration. And we focused on an analysis between the two colored bars of the first 3 seconds. Actually, I'm going to show you in the first 2 seconds, because that's where all the action was, of inspiration. Because we want to see what's happening during inspiration of this mechanically ventilated breath from APRB. The red line is a digitally filtered signal that's taken out the oscillations and just leaving the ventilator waveform. So when you subtract the red line from the black line, we get the oscillations. And the way we did the analysis was, well, we calculated the impedance over between those two bars using a 1 second sliding window. So 1 second is the repeat period of the oscillations. And we slid it across that data set, calculating impedance at every position. And the positions went at 128 hertz. So we get a continuous, if you like, a continuous progression of impedance with a resolution of 1 second sliding over that window. And then I fit the simple single compartment resistance, inertance, elastance model to each impedance. This is the equation of it here. So I end up calculating resistance, inertance, and elastance of the respiratory system continually throughout that period. So they are now functions of time. And so these are the results that I want to show. Over the first 2 seconds of inspiration, we've got the resistance, the elastance, and the inertance of the respiratory system as functions of time. And for the 4 different groups shown by the colors, the most injurious group is in red, the least injurious group is in black, and the sort of intermediate groups are green and blue. And you can see they separate out quite nicely. By the way, this is the average plus or minus standard deviation of a lot of impedance measurements average from multiple different time points, but they all come from the groups that I showed. That's why the error bars or the confidence intervals in terms of standard error are quite small. So the first thing I want to point out is that the resistance shows a peak in the most injured group at about half a second. Whereas if you look at the other 3 groups, there's a suggestion of a knee in the curve in the blue, which also has excessive recruitment and de-recruitment, but no overdistension. There's a little bit of a knee there, which occurs at the same point as in the red. The other 2 groups, which had short expirations and therefore limited recruitment and de-recruitment, don't show that peak. They have a possibly a peak right at the start there, and then they're monotonically decreasing. If you look at elastance and resistance, they're doing the opposite, and they're noisier because a couple of reasons. The resonant frequency of these pigs is about 6 hertz. Most of the data is above 6 hertz. So we don't get a very good fix on elastance. And also resistance and elastance both come from the imaginary part, whereas resistance is the only thing that comes from the real part. So it's statistically more robust.
So I'm going to undo my ignorance, because most of my group, when we're working in the time domain, we're thinking of a single frequency, but you're not using a single frequency here. This is full spectrum.
Yeah.
So just educate me how that happens.
Well, what I'm doing is I'm calculating the full spectrum, but I'm fitting a model to the impedance spectrum. It's a very simple model. It's actually, I'm taking the average over all those frequencies of resistance.
Oh, okay.
That's effectively what it is. I could fit a fancier model, 2-compartment model, but I want to just get a global overview of what these properties are doing.
Have you ever looked at it using a single frequency at a time? Does it change it at all? Or?
I haven't looked at it here. I mean, but we could have done if we were only interested in resistance, we could have used a single frequency. The problem with that is you can't separate out elastance and inertance. You need multiple frequencies, or at least 2 to do that. So we see this peak. Now, how do we interpret this? And this is the last thing I'll say. What I think is going on is that when you have a longer expiration, which is what happened just before the red guy, the lung actually starts to de-recruit, because de-recruitment takes time. It's not just a function of pressure, it's a function of time. It starts to de-recruit. Then you start inflating the lung. In the initial part of inspiration, the de-recruitment continues, so the resistance goes up. Eventually, you get enough volume into the lung that it starts to reverse it, which is where the peak is, and then it turns it around. What we see in the much shorter expirations, shown in the black and the green, is that we have possibly a peak right at the start. So we had just caught the beginning of de-recruitment and stopped it in time. So I think this is pretty strong evidence of what we are suspecting is the case, that by having expiration short enough, we don't give the lung enough time to actually start de-recruiting, so we have to rip it apart. What's happening in the red guy is the lung is being ripped apart at this point, and that's very damaging. So in conclusion, oscillometry can be applied during mechanical ventilation for anybody who's interested. Respiratory system resistance decreases monotonically during inspiration when APRV is short, but not when it's long, because of the reasons that I've just said. And what we're thinking is that oscillometry might be used to titrate, in any particular case, what the perfect expiratory duration is, to make expiration as long as it can be. Expiration as long as it can be, but not long enough to have intratidal recruitment and de-recruitment going on. And thank you very much. I'll be there.
Any questions from the room first?
So Jason, really, really nice work. I gathered from the associations with the histology that, and the mechanics that the index that's best predictive of damage is resistances. Did I get that right?
Well, it is in this particular case, and I think it's largely because we got the best signal to noise ratio and resistance. We also, we had a trough in elastance,
Yeah.
which would indicate.
Do you mind joining?
There you go.
Oh, sorry, yeah. We had a trough in elastance, which was the sort of converse. Normally, I would go for elastance to look at this signal. It just turns out that resistance had a better signal to noise ratio.
But if that's true, then when you go to apply this in the clinic, that would simplify the instrumentation, correct? Or do you think it matters?
It wouldn't simplify the instrumentation, but it might simplify the analysis. I don't know.
Or the understanding?
Yeah, it's just, this is just
Yeah.
where we are at the moment. I mean, to actually do this in the clinic, you'd have to have all that crap in there. In a smaller version, hopefully.
Why did you pick the particular time duration of the expiration? Just out of the blue, or did you have?
No, not out of the blue. My colleagues have a technique. Our group works closely with an intensivist in Maryland called Neda Habashi, who's a big proponent of APRV. He puts his patients on it all the time.
Yeah.
And the way they choose T-low, as they call it, is they terminate expiration when it has fallen to 75% of the peak value right at the start. And interestingly, you're sort of walking the knife edge here, I'll tell you, because we've also compared this, and they've compared it to what happens if you terminate it when expirations fall into 25%. So expiration's a bit longer. And that's actually how these times were chosen. And you go from being very protective to utterly disastrous with an increase in expiratory duration of less than a second. So, my friends here are desperate to have some kind of a clinical trial to test this out. But, if you have a clinical trial, a multi-center trial, where less than a second is the matter between success and disaster, that's a challenge, put it that way. Hopefully they'll do it, but yeah.
So a couple more questions from the room first, and yeah.
Yeah, so Jason, coming from pediatrics, we use high frequency oscillatory ventilation where we fix a pressure usually in the 20s.
Yeah.
And we oscillate at a fixed frequency, usually somewhere, it's usually 8 to 10 hertz. And it's very good both for oxygenation and ventilation without causing damage. Charlie Bryant, like 40 years, showed how this was actually quite protective. And there are some units, particularly in Europe, who that is their standard as opposed to a rescue in North America. It's more of a rescue. So I'm just curious, when you added an oscillation, especially if you picked a particular frequency, whether you could forego the exhalation and get adequate oxygenation and ventilation.
Yeah, that's a great question.
It's been tried in adults. The problem is that they found that they couldn't get a speaker that would generate enough pressure, oscillations, that it would go far enough down into the airway.
Yeah, I mean, that
In babies, you put your hand on their thighs, it's vibrating. I know we're getting.
that can be dealt with by linear motor oscillations, kind of thing. But actually, to address your question, there seems to be a difference between kids and adults. There have been clinical trials of high frequency oscillatory ventilation in adults. None of them have been shown to reduce mortality, which is curious because if 6 mils per kilogram is better than 12, why not none? Why wouldn't they be better than 6? And it turns out that they have not been successful for this all-important reduction in mortality. Nobody really understands why, but it's fallen out of favor in adult ARDS patients. Another thing about this APRV, and I'm not evangelizing here, I'm just telling you what the thinking potentially is that extended time at a high pressure, provided it's not too high, actually over a long period of time will recruit the lung slowly. Because if you have a consolidated lung, what you want to do is open it up as much as possible so that you don't have actually really any closed units, because the interface in the tissue between closed and open units is a very high stress region. So if you can eliminate those, and what Gary Neiman and Neda Habashi, sort of very keen on this method, have said that Apparently, in a lot of patients who've been successfully done with this, you can open up the lung over a period of hours or even days. Why did you never take them off? So you gradually open the lung up and you keep it open. And so that extended high pressure.
I think one of the reasons, I mean, in infants, usually dealing with basically surfactant washout type of model, which is what your model, whereas in adults.
Yeah, this isn't the
an
greatest.
sepsis type of model.
This isn't the greatest ARDS model for that. So we're actually going forward, we're going to do other nasty things to them.
Any other questions from the room?
Hi, Jason, really nice. Two things. Was there enough time from the injury to being on the ventilator and starting the protocol for inflammatory mediators to start circulating, basically getting a cytokine storm and having that portion of the ARDS actually happening in these pigs?
I would guess not. I mean, we did blood analysis and found that those things were elevated, but I would guess probably not. So it's a pretty limited. It's very clean and easy to do, which is why we started with this. But it's you could say from a clinical relevance point of view, this particular model has some shortcomings.
Yeah, because I maybe I got it completely wrong, but you had resistances rising. Well, they rose. One of them rose and then started to fall right throughout that pressure hold period. But the elastances were rising, weren't they? They were doing the converse.
They were going down and then going up.
Yes.
Yeah.
Why was elastance going up and resistance coming down at the same time?
I think it's something to do with an interaction between the strain stiffening of the lung and the de-recruitment. I have to think about that a bit more. Actually, one thing we did very quickly is
Usually elastance going up is de-recruitment, and you have elastance going down and you're still.
well, we also fit time domain models for non-oscillatory data, which I didn't show. And for those most injured pigs, the dynamic pressure volume relationship was much flatter than in the non-injured pigs. And the reason, I think, is because as they were being inflated, the lung was de-recruiting while you were getting strain stiffening. And those two effects offset each other when you end up with a flatter PV curve. So it's a bit complicated.
So the lung getting stiffer, opening the airways.
Yeah, but I will say one final thing. One of our collaborators in the study was Dave Kaska, who is a very technically competent biomedical engineer, but also a board-certified anesthesiologist and critical care physician. And he's told me, somebody whose opinion I value greatly, he's told me that APRB like this works really well in some patients, those who are recruitable, but not every patient. And I think, that's the way to look at it. You've got to choose your patients.
Did you want to comment?
Oh, I was just going to comment on the strain stiffening versus resistance thing. We've seen the same thing in mice.
Yeah.
So I think it's fine. It makes sense.
That's not an outlier. Not a new idea. Other people.
Yeah.
Yeah, thank you. So what only say to say that a PRV, we use it often in our department in the hospital, medical hospital. So my question is, usually we use it only to recruit, but usually we adjust only the T high and the P high.
Okay.
It's rare we use the T low. So how about exactly? So usually we use the T high between 3 seconds and 4 seconds. Sometimes with the COVID, so we went to 5 seconds. So how about when we adjust 5, 4 seconds?
I'm not sure I can give you a totally authoritative answer on that. My opinion, at least in this model, is that that's less important than the T low. In fact, the T high varied quite a bit among the animals, from about 3 seconds out to 5 or 6. I'm not sure that made too much of a difference. I think the important thing is that most of the time it's at this high pressure, and very briefly you have these release times. But it's the release time is really critical, because if you allow enough time for those things to come, alveoli to come like that, now you've got the Velcro effect repeating apart, and it's a lot of work both in these pigs and in mice shows that that's really damaging over extended periods of time. So we're trying to find ways to avoid that.
Did you look at surfactant function at all in these? Or
No,
okay,
obviously,
because I think that's one of the driving factors with that collapse and reopening is it physically destroying the surfactant layer.
Yeah,
That
sure.
like showed up in the mice when we were running peak zeros.
Yeah,
High pressures.
Yeah.
That's what tween does.
Yeah, that's right. Tween
Kills
destroys effect.
actually all proteins do. So if you get protein leak, albumin leak, like you see in sick people, that's what's killing the survivor.
But the sort of the fundamental physiological process underlying all of this, which we've done a lot of work on over the years, starting with Gil Allen kind of stuff 15 years ago, is that recruitment and de-recruitment, commonly thought of as depending on pressure, but they don't just depend on pressure, they depend on time. So if you have a lung at some pressure and you suddenly drop it, it's not like you suddenly de-recruit to the level of that pressure. The de-recruitment will take time. It takes time to manifest because you need time for menisci to go up across the airways. You need time for the alveoli to close. And you can characterize that by looking at the dynamics of elastance as a function of time following a maneuver like that. So what we're trying to do here is exploit the time dependence of the recruitment.
Okay, Zoli, you take the last question here.
Yes, thank you. It will be short. Congratulations, Jason. I have a very minor remark, in fact. You are measuring airway opening pressure probably at the end of the proximal end of the AT tube.
Right.
So in the phases of fast inspiratory and fast expiratory flow, there's an additional huge pressure drop or pressure increase, resistance increase, which is probably with this 1 second window you embrace and maybe expand these transients. Do you have an idea how much it's confounding the resistance readings?
Haven't really looked into it. It's a great question. I think it's probably confounding things more during expiration. I didn't look at expiration because to get signals like that, we have to do ensemble averaging of a lot of signals. And the expiratory duration was actually different in those various pigs because of the way we were choosing the time to cut off expiration. So the ensemble averaging didn't work very well. But I did try it a bit, and it was harder to interpret what was going on. During inspiration, you have a sudden increase in pressure, but the flow isn't instantaneous. The flow actually takes a while, I don't know how long, some fraction of a second, but it's not an instantaneous development. But it's a good question. I mean, I didn't give you a very good answer.
Thank you very much.
Thank you.
So I'm not being too strict with time control, just so that so welcome Andy Clement from the University of Brussels in Belgium. You'll be continuing the theme about ion median low frequency oscillometry.
Thank you. Share it.
Let's share the green at the bottom.
All right.
Good.
Everything's good?
Yeah.
Oh, great. So I'm a PhD student at the University of Brussels, that's the engineering faculty. Nevertheless, I'm still going to present some clinical results. So that will be, for me, a bit on a slippery slope. Nevertheless, the principal investigator of the clinical trial, Professor Dr. Joop Jonk here, is available in the audience to catch any mistakes. Now, that being said, what I will be talking about is about low-frequency respiratory oscillometric monitoring during mechanical ventilation of intubated patients. So context is really about monitoring of these patients via respiratory mechanical properties, more in detail, the resistance and the compliance of these patients. Resistance and compliance, because that's what the ICU physicians are used to, But the problem we are looking into is that the standard of care measurement tools are of invasive nature and or require patient passivity. And when I'm talking about standard of care, we are looking at a static compliance estimate via a low flow PV loop or a dynamical compliance and resistance estimate via the fitting of a differential equation to the ventilation waveform. Now, at the bottom, we can see a graph who shows a typical treatment procedure of a patient at the ICU, but it can differentiate a lot. But over here, I display the patient in the beginning having some controlled ventilation. And in that situation, you can have a statical compliance estimate. You can get a dynamical compliance estimate quite easy with the current technology. However, usually they will go towards more supportive ventilation strategies. And in that case, the dynamical compliance estimates are failing because of patient activity that intervenes with the estimation protocols. So the solution we are looking into is, can we use hospital oscillometry to actually get a compliance and a resistance estimate in as well as the control as the supportive ventilation strategies, such that we could monitor those parameters over the treatment. And we are going to do that by the application of a low frequency, but low, I mean like really low, between 0.13 Hertz even to 5 Hertz, with a multi-sign excitation signal. Now, last year I was also presenting, and then I talked into the technique of that we were using and how we developed it and the details of that. But now I really will look into the application of this technique in clinical practice and how it is performing. Now, however, the clinical trial is still ongoing. So we're still doing the measurements. We are now at patient 19 of 21. But I will present results of the first 10 patients where we have an interim analysis just to see like if you are on track. So it also means that some of the analysis still needs to be performed, but however, I think it's a great opportunity to already get some feedback on the ongoing work and improve the data analysis afterwards. Now, let's fetch a summary of last year. So last year, I presented the technique, but on emulator. And there we could see, okay, the resistance and compliance, we can get this estimation during mechanical ventilation, and it's comparable with the standard of care. And then the beauty of the technique is in the how. How did we do it? Via spectral separation. That's in short, what does it mean? Well, it means that we'll, in the frequency spectra, we will look at where we can place our frequencies to excite. So the common choice is to use the 5 Hertz to avoid the total disturbance of ventilation. But however, if you would look at the spectra, you can see not all frequencies over there are as much contaminated by the ventilation disturbances. There are some spots where the noise is low enough that you could put some energy into and still get a decent result out of it. However, some further things need to be considered to still get to a compliance and resistance estimate that is useful. What, for instance, we do is, for instance, we measure 2 minutes. That's sufficiently longer than, for instance, the 10 seconds of base, but it also allows to have a large averaging period to still get these 2 parameters of a decent quality. Now, because he also very beautifully explained how his setup works. Well, relating to that setup, we are working with a turbine. The turbine will provide the mechanical ventilation, but the signal that we send to the turbine is not only the ventilation waveform, but is the ventilation waveform superimposed with our excitation signal, this small perturbation on top of it. And it's a turbine that tries to create this signal and has a decent result. However, what we see by using this turbine that is optimized for ventilation frequencies, we see actually a good performance at the low range, so like around 1 Hertz, and a worse performance at the high range, the 5 Hertz. So therefore, when we are looking at like the smart excitational design, we need to take the performance losses at the higher frequencies into account to still get a decent result. However, I can dive more deep into it, but I will skip that to the questions if they are about this technique. Now, at the end, what we saw, okay, so with this avoidance, we can get a patient-friendly estimation that's less disturbed by simulated patient activity. Simulators, because we are still talking about emulators. However, an emulator is very nice. It's very periodic. It's definitely not comparable with like patients then get the data much messier. So therefore, an equivalence trial was being designed on 21 intubated patients, where we will compare the compliance estimate of standard of care with the compliance estimate of a RO via a prospective equivalence study. What we will do is we will take the compliance estimate of standard of care as the ground truth, and we will look if our compliance estimate of oscillometry is in between a 10% interval. If that's the case, then we consider it as equivalent. Of course, that's the primary objective, and that's where we did our power study to get to the 21 patients. And the 21, for the 21, we will do 6 measurements, 3 in the morning, 3 in the afternoon, and then we will analyze this package of data to see if this patient has equivalence between oscillometry and standard of care. However, we're also looking at the resistance, and then the secondary objective is maybe the more interesting one from scientific curiosity perspective. We will also see if we can use this measurement procedure to get this compliance resistance estimate in the pressure support case, which would be a great benefit. However, currently there was no comparator in this ventilation modes, so we're just looking at, is it still a reasonable estimate? Is it feasible to perform? If yes, then future work can focus on this comparison in this ventilation mode via, for instance, using the esophageal balloon. So interventions we are performing is just mechanical ventilation via a modified ventilator, the DENCARE. We will perform a PV curve measurements to try to get the ventilation into the linear region of the respiratory system, and we will apply our oscillometry measurements at the 5 Hertz as well as our low frequency oscillometry. Over here, I provide some summary of the inclusion and exclusion criteria. It's not every inclusion and exclusion criteria, but the most important, the most stringent criteria are listed. So we are looking at intubated patients that have controlled ventilation for at least +4 hours. So it already gives us like the more critical patients with worse outcome perspective, given the fact that the doctor is assuming that you will have some long time on controlled ventilation. It's hemodynamically stable and survive for at least 48 hours, and protective ventilation, which is a PEEP less than 10, a delta pressure of 15 or lower, and fraction of inspired oxygen less than 60%. So exclusion is the assist ventilation, because in that ventilation strategy, we currently have no comparator. That would not be very useful. No ECMO, no ventilator asynchronous. And of course, the ventilation should be possible above the lower inflection point and below the upper inflection point. So setup we are using is a mechanical ventilator on the left with some minor modifications so that it can apply very decently the oscillometry signal via press on the button. We will insert the excitation signal design specifications via USB stick, and the design itself, the excitation signal happens on the laptop. How we design is first we make a ventilation measurement. We go to the laptop, design our excitation signal, go to the ventilator, perform the measurement, which we can stop at any time. And then we get the data, we can analyze it automatically to verify if we have these parameters at a decent quality. So as I mentioned before, we're doing 6 measurements a day, and we're using this day for our primary objective. With all these estimations being performed, 3 in the morning, 3 in the afternoon, and we have 4 hours in between the first measurements of every session. Now, if the patient continues its treatment, we will also measure up to 5 days, but then the patient can be switched to pressure support when medical the medical site determine that it's a good time, and we will just continue the measurements. But of course, in pressure support, we are not going to measure a statical compliance, we're not measuring a dynamical compliance. It will just be the 5 Hertz and the oscillometry that we will perform. Now, let's take a look at the results. Over here, I show the non-parametric impedance estimate of a patient during pressure control. And with the parameters resistance and compliance being shown here, accompanied with their coefficient of variance, and we can see it's pretty it's a quite low percent variance, well below the 10%. So in this case, we had a decent estimate. We also estimated the inertia, and it's comparable with the model that already Bates showed. When we just look at how well we succeeded in squeezing the exchange signal into the spectra, well, this graph shows the pressure spectra over the frequencies. It's the pressure, but it's in the decibel scale. So the main message I want to convey with this picture is we can see exchange signals as the purple dots, and they are below the blue dots, which is mostly the noise, but you can also identify the ventilation harmonics, which are a bit above. Over here now, I'm showing the same impedance spectra of the same patient, but I think it was 4 hours later, they switched to pressure support, and you can all identify already like a higher uncertainty at the higher frequencies, but you're still getting like a decent estimate. And when we are going to look at the impedance spectra, you can already see that these very identified ventilation harmonics are gone, because there's more leakage, there's more frequency variation going on, but still our algorithm is able to identify the spots which we can excite. And again, the excite frequencies are indicated with the purple circles. Now, let's look at our primary objective, which was equivalence for the pressure control case. On this graph, we are displaying respiratory compliance of oscillometry on the y-axis and respiratory compliance comparator on the x-axis, its value, and then we also show the standard deviation for the parameters, and the line of identity is being drawn through the patient data. Except patient 2 and 6 is excluded, because over there the estimation was performed in the nonlinear region. And therefore what we definitely saw, especially because our modeling strategy as well as from this standard of care was based on linear assumption, and therefore in the nonlinear region that didn't work quite well. So therefore those 2 data points are excluded. And then over here you can see the line of identity drawn through, and you can see that we have these equivalents with the line going through, for instance, the 60 point over here, and like the 40-40, which is of good quality. And also when we perform a 2 one-sided t-test, we are getting a p-value well below the 0.05. One thing more I want to mention is on the previous slide is that the coefficient of variation for 35 out of the 40 was below the 10%. Of course, we also had the resistance being estimated. Well, definitely what we could see for the resistance that in general our respiratory resistance of oscillometry was like 80% of the dynamical estimation. So over there we don't have any equivalence, but it's can be considered as a bias that's comparable throughout different patients. However, also the uncertainty significantly increased for our resistance parameter. Now, let's get into the more interesting objective, the secondary objective, where we were looking, okay, now we have this protocol, can we actually use it to monitor? So what we are displaying here is the data of one patient with every dot being the mean of three measurements at that point in time. And we're doing it for blue is the comparator standard of care estimator, and the orange is our oscillometry protocol. The standard deviation is also being plotted on every dot. And in the beginning, we had the issue of the lower inflection point being greater than the PEEP, although it was a minor difference. However, those points should be taken with care, because maybe we have this non-linearity that can play into a bad estimation. However, next to it, we have 2 points with a decent quality, and we can see that the orange, the oscillometry, is very close to the blue standard of care. So we trust that value for the compliance estimate. Next to, we switch to the patient switch to the ventilation mode, where he can have control ventilation, but the device will assist his breathing. And there you can already see the standard of care, the blue one, jumping up. to which is quite strange given the earlier estimates, while the oscillometry value is still in the same range, which is interesting. Then we switch to pressure support, and over there is then the domain where it can get more useful. The standard of care wasn't able to get as any estimates, of course. However, the points being shown are those ones of the oscillometry estimator. You see clearly higher variance due to patient activity. It's not a periodic ventilation any longer. And in this patient, on day 7, they return to controlled ventilation. So that is a very interesting situation, because then the endpoint is again in controlled, where we have our comparator. So to be very precise, during pressure support, we cannot say if the displayed estimates are the true value. Currently, we cannot do, but this data already shows that it's interesting enough to continue towards a comparator study in pressure support with an esophageal balloon, because those estimates seem to be in a logical range when we're looking at the controlled dots over here and over there. When we're looking at the resistance, we have so the bias that I explained being visible in the controlled case, as well as in this medial case, and then an event that's quite interesting is over here on day 5. We saw it's increasing, and not only the mean value, but also its variance was rapidly increasing. Now, on that was started on Monday, but on Tuesday afternoon, they performed a bronchoscopy and identified inflammation at the upper airway. After like a day after, the patient has a septic shock, and they returned to a controlled ventilation, but also performed some treatment to get the inflation to control the inflammation. Let's put it that way. So that's quite interesting. It's, let's say, promising. However, it's still hard to link this event to the data, but at least it shows something that's interesting to investigate into the future.
Did they know something was happening clinically when your signal became abnormal, or did your signal pick it up before the clinician?
Well, I would claim it picked it up one day before, because it's only from the first green dotted line that the bronchoscopy showed the inflammatory reaction in the patient. Whereas one day before, already saw this happening, but definitely we didn't show this data to the doctor because it's still an explorative protocol. So we're not influencing the medical trajectory of the patient, because it still could be that it's just like, I mean, bullshit. I mean, it's not verified. So therefore, we keep it on our side, and maybe this can be interesting. So, but let's put it that way.
On a very practical level, you know, you don't do a bronchoscopy immediately, because you still have to hold the feeds and then organize the bronch. So it isn't, so there's at least a window, sort of from a very pragmatic sense, of at least, you know, 6 hours. And when I see a patient at 8 o'clock and I say this patient needs to be bronched, it won't happen until the afternoon, right? Because you, so I think that, you know, probably whatever that event was that triggered the clinical decision happened before your line.
Yeah, well, definitely. I think the main advantage is just it picks something up and it's non-invasive. It can be used as an indication, but indeed, if it would relate to any patient outcome, I'm not going to say anything about that. That's future work, definitely. So let's get to the conclusion. So where are we now? Well, in the controlled ventilation, 8 out of 10 patients had statistically equivalent compared to the dynamical compliance. And we could see that our low frequency oscillometry is feasible in pressure support. However, future work, we still we're not sure that the compliance of the respiratory oscillometry is equivalent to the standard of care in pressure support, and therefore we will need to perform measurements with the esophageal balloon. Definitely some further technical improvements are needed to reduce the variance, as well as to get the bias in the resistance estimates gone. And then next, we still are not having any like patient-related benefits relating to this monitoring principle. That was it from me, and I'm happy to answer any questions that there may be.
So in usual fashion, we'll start in the room with any questions. Right.
Maybe, Andy, I'm retraining myself as a clinician who does not manage ventilated patients, but the oscillogram you showed of your representative patient, it was resistance and reactance as a function of frequency that you showed, right? And you had a resonance frequency that was under 5 hertz, which is what I don't see in my patients. Is this because the mechanics of the patient plus the respiratory ventilator that they're on gives you different mechanics than if I was just doing patients in my office?
Well, very good question, and I doubt I will be able to answer why this inertia is so low. It was not only for this patient, also other patients are showing this behavior. Maybe the more
Related to that, where do you measure pressure and flow?
where it's right at patient 9. We do it with a pneumatograph similar to the one you showed.
Okay.
No, so it's
Yeah, well,
the healthiest.
like yesterday there was this inertia discussion, and I was like really looking into it, and I was like, okay, we have it quite early, and then I was looking, maybe is it just like obesitas with this high weight that need to be accelerated? Maybe this can play into account, but I'm totally not sure.
I was simply thinking that's the healthiest intubated patient I've ever seen. I'm hoping that maybe some of the people online or in the room can explain.
Now, one possibility is we found pretty low resonant frequencies in our pigs. And I hadn't thought about this before, but it could be related to the endotracheal tube because you have an extra column of air, which is increasing your inertance. And if you do that, it will drop your resonant frequency. So it could, that might be.
Also,
Your that's due to just actually at the ventilator, right?
no, it's at patient mouth. Yeah, but that definitely can. Maybe the thing is currently we're not removing the impacts because Pascal had a very good paper about it to reduce the impact out of the impedance estimate. Currently, that's not performed. So the impact of the endotracheal tube is still in the measurements. So, but the thing is, we cannot remove it because our comparator also doesn't remove the impact of it. So, endotracheal tube will have an influence, and maybe indeed that's the idea.
So you're looking at resistance throughout the cycle or just during inspiration?
Well, throughout the cycle, throughout the 2 minutes.
But then you get Bernoulli effects of when you because of the differences in the diameter depending on the relative size of the tracheal tube to the trachea. So if it's tight, then you won't see as much. Whereas if there's a big difference, then you're going to get. It doesn't matter whether you're going in or out.
I would think the way to correct for it, what I would do, and you could do this both for your comparator as well, is characterize the pressure flow relationship tube. Subtract that from your primary signals in the time domain before you do the impedance analysis.
But that doesn't work. Zoli and I had grappled with this because before COVID hit, Zoli, Stuart Godfrey, who was online yesterday, and Peter Goldberg, we had done 3 patients, excuse me, in the ICU with a 10 hertz mono frequency in line. And what we really had wanted to get was an endotracheal tube that would allow pressure or flow to be measured at the end of the tracheal tube. And there used to be on the market tubes that had a cannula down. The problem is clogging and such, but we could never get our hands on. Now, I don't know.
You also have to extend beyond the.
Right.
Steve Loring studied this years ago. He's a well-known anesthesiologist. And the problem is it makes sense that you would measure it and then subtract it, but it doesn't work because of the diameter.
No, but Charlie, if you characterize that nonlinearity, in fact, the reason why you should do it this way is because the relationship is nonlinear and it is direction dependent. So you subtract that direction dependent nonlinearity in the time domain before you do any frequency domain analysis. So you're effectively taking out the endotracheal tube and all the nonlinearities and everything associated with it.
But the relative size of the tube to the patient trachea varies from patient.
Well, you have to do it. You probably have to.
So that's why we
characterize.
would put the pressure tap beyond the end of the trach.
No, I understand what you're saying.
Yeah.
But it's a question of degree. I mean, no correction like that's going to be perfect. The
No.
question is, did it get you close to where you
Yeah.
are?
Yeah, and then we could effort to actually put a catheter down inside the tube. It's,
But then you gotta keep it clear. That's the problem. What kind of patients were these? The ICU or just in the surgery?
It varies. It varies.
Well, that may explain why they're,
Yeah, they were helping.
Yeah,
because they were just getting surgery.
but yeah, there are some patients. Maybe Professor Jonker can answer it, but definitely there was some surgery patients. There were also some COPD patients. Just based on like, do you think you will be there for like more than 4 hours? Is he stable? Does he has a linear region? So that already exclude like the very worst patient. So that was a bit of selection, but indeed they can be included.
Yeah, you may have explained this and I missed it, but the impedance of the lung below 1 hertz is quite frequently dependent due to the viscoelastic properties of tissues and the heterogeneity as well. So you were comparing some low frequency resistance or compliance to what you measured from the ventilator. But what frequency were you, when you calculated your impedance values of resistance and impedance, were you matching it to the ventilation frequency, the fundamental frequency of the ventilator?
Matching in some sort, yes, because we are squeezing our excited frequencies in between and using the same, like the ventilation waveform has a spectra between 0.1 and 3 hertz, and we're choosing the same spectra. And it's because we are choosing the same spectra that our estimate based on our excitation frequencies can get similar.
So it's similar, I see.
Yeah.
So how do you do the analysis? Do you use some kind of a comb filter to get rid of the ventilator waveform and just leave the oscillations?
It's in a frequency domain, and we look at the excited frequencies, and then we will perform an analysis on that. And then it's a weighted linear least square cost function minimization we are performing on those excited frequencies.
Okay. When you calculate the FFT of your data,
Yeah.
what's the length of the signal that you're doing that on?
Well, yeah, it's 2 minutes.
2 minutes.
Yeah, yeah. And it's a one. So we do not averaging, because what we need to ensure is there is a spectral separation between the ventilation and the excitation signal. Now, ventilation, the physician can choose like value to like 2 minutes. So like for instance, 10 breaths per minute, 20 breaths per minute. So it's 60 seconds. That's the interval. So what we do is we have our excitation frequency being 40, and then the total measurement time is 120, so that ventilation frequencies are on even lines, and our excitation frequencies are on odd, so that they are separated and don't influence, so the ventilation frequencies don't influence our excited harmonics, so that we can use those excitation frequencies to compute our model, our RNC, those two parameters.
Okay, I see what you mean. Just a minor technical point though, that how do you deal with edge effects? Do you window the thing before you do the FFTs with like a Hamming window or something? Because you don't have an integer number of breath cycles and oscillatory cycle.
Yeah.
Then you're going to get leakage effects, causing a blurring of the spectra.
Well, there will not be any leakage effects if you cut your data in 120 seconds, and the time window of excitation is 40 seconds, and the time window of your ventilation is 60 seconds. Because then you cut integer windows.
Right, so you did what I was thinking.
Yeah, exactly. Yeah.
Fair enough.
But there will be some leakage just due to the imperfectness of the device. In the data I showed you, they were a bit too messy to really show it. But if last year there was on emulated data, and you could really see the leakage scars. Just it isn't perfect. Sometimes we also lose a sample. There is also other device performance that we would like to see more ideally, but it still creates a bit more.
I see what you did. That's nice. Yeah. Thanks.
Anyone online? No, I don't see any other questions. Okay, thank you very much.
Oh, we had one.
Oh, we do. Is that on the chat there?
That is a finger raise. No.
Thanks, guys. Yeah. So I'll just read it for you. Jason, nice results. Thank you for sharing. The 1 second sliding window is larger than the brief expiration. Oh, this is, yeah.
Oh, that's my.
Yeah. Let's blame that one on the moderator.
Hi, Jake.
Perfect. Thank you very much. So we're going to switch gears, talking about respiratory symptoms, functional status in the post-COVID population.
Sure.
My name is Tadehisa. I'm a respirologist from Japan and currently training as a clinical research fellow to learn oscillometry. I'm greatly honored to have opportunity to present and discuss our research study association, of oscillometry abnormality with functional status and respiratory symptom post-COVID. About 10% of the patients reported long-term symptoms after acute COVID infection. The prevalence of symptom is disproportionately higher than the normal objective metrics such as spirometry. Earlier studies find that the patient with post-COVID showed restrictive defects with reduced DLCO and exercise capacity that improved at 1 year. However, there are some persistent physiological impairment. And recently, hyperpolarized xenon MRI and expiratory CT identified ventilatory defects and small airway dysfunction in these patients. But the roles of oscillometry is not well established. So the purpose of this study is to investigate association of oscillometry abnormality with functional status and respiratory symptoms at 6 months post-COVID. We utilized the cohort from CANCOV enrolled between August 2020 to March 2022. The date of infection is defined by date of symptom onset or positive test. Admission criteria is supplemental oxygen for ward and ventilatory support for ICU group. We identified 246 subjects as eligible for the analysis. Outpatient group was younger and less BMI and less comorbidities and complications. However, there was at least one respiratory symptoms in more than half of the outpatient group, with trend to increase with severity. Spirometry data was almost normal across the cohort. However, abnormally low 6-minute walk distance is observed in 21% of the outpatient group, with trend to increase with severity. Spectral oscillometry showed that R5 is not different between groups. However, R5 to 19, X5, and AX are significantly different in outpatient and ward ICU group. And the Venn diagram showed prevalence of abnormal findings among the overall cohort. Restrictive ventilatory defect and obstructive defect is defined by these scores, and abnormal R5 to 19 is defined by the metrics more than 0.8, and abnormal AX defined by the these scores. Oscillometry detected 67 patients with normal spirometry and abnormal oscillometry. Linear regression analysis identified that abnormal metrics were significantly associated with clinical outcomes in these patients. We next conducted intra-breath oscillometry. As you already know, intra-breath oscillometry is measured by monofrequency and tracks resistance reactance during each breathing cycle. REE and REI is end expiratory and inspiratory resistance, and XEI and XEE is end inspiratory and expiratory reactance. Intra-breath oscillometry showed that RE resistance values at each end of expiration, the inspiration is significantly different in each inpatient group and outpatient group and inpatient groups. And also reactance values is significantly different between in outpatient and inpatient groups. The difference between end expiration and inspiration in reactance and resistance and reactance is most prominent in ward group. This is correlate regression analysis. There are many parameters associated with 6-minute walk distance and post-dyspnea score. As you can see, the difference between end expiration and inspiration of reactance value and XV prime. Those parameters are known to be a marker of tidal expiratory flow limitation is significantly associated with clinical outcomes. In conclusion, oscillometry is more sensitive than spirometry in detecting abnormal lung mechanics, and intra-breath oscillometry is more sensitive than spectral and highly correlated with functional outcomes and respiratory symptoms at 6 months post-COVID. I'd like to acknowledge these funding institutions and our lab members. Thank you very much.
Okay, any questions from the room? Right.
Very nice. Congratulations. Maybe it's me and you'll just say the postdocs don't show any difference, but were your ward patients sicker by intra-breath mechanics and spectral than the ICU?
I'm sorry, one more.
Were the parameters more perturbated or worse amongst the ward patients compared to the ICU? Because clearly I would have thought the ICU should be sicker.
Yeah.
And happily the ward, the non-admitted people seemed relatively okay.
Statistically, there is no significant difference between ward group and ICU group, but in spectral measurement, the value is higher in ICU patient. But intra-breath oscillometry is not so different.
So on the post-talks, the ward and the ICU didn't show a difference, even though the numbers looked like they might be higher.
Yeah.
Anyone else in the room? Oh,
Really neat. So I may have missed it, but this is 6 months after they've got COVID.
Yes.
So they're in the ICU or they're in the ward or they're outpatients when they were measured 6 months later
6 months
due to COVID.
after discharge.
After 6 months after discharge.
Yes.
But some of them were in the ICU, some of them went through the ward, some of them were just open. I get it. So But they still have significant, or you had a significant number with retained respiratory symptoms.
Yes.
Yeah.
More than half of the patient has
Yeah.
symptoms. Yes.
So in a few minutes, I'm going to present milder
Oh.
patients because I don't have any. Thank you for that.
Yeah, so one of the things we know is that patients who do spend time in the ICU end up with a restrictive defect that
Yeah.
often persists for many years and actually doesn't resolve. Now, this is due to other things besides COVID. So I think the thing that's really significant here is, as Ron was pointing out, that the fact that if they go into the ward, that they're much more likely to be ill out. We've done a much smaller study, not as well done as yours, and those that had been were not hospitalized, and we don't see much of a difference at all, actually. The lung function is pretty well preserved. So wondering about, as we know, COVID has many variants, and the earlier variants were much more disastrous than the current ones.
Yeah.
So do you have any idea about what particular variant these folks were?
In this cohort, I think these are alpha to in the middle of Omicron.
Right.
And this study, we stopped this enrollment because vaccination is widely.
Right.
So I think after this, our
So this was
introduction
of vaccine. So this would have been some of the early variants.
Yeah,
that were
yeah,
much more severe.
exactly. Yeah.
Got it.
We had stopped enrolling for that very specific reason. Once people got, then we knew that it would be a complete.
Huge confounder. Yeah.
So Brian, we should have persisted and done.
I know.
the study we wanted to do that we couldn't get through ethics on.
They're still proving it. It still works.
That's right.
Pending, every time I go,
Conditional,
well,
conditional, yeah. From the online chat, is that Dr. Rabinovich?
Yes, a great talk. Just a question and a comment. Could you tell me the difference physiologically between intra-breath oscillometry? What is it showing compared to spectral oscillometry? I'm not familiar with intra-breath oscillometry, number one. And number two, the comment that we did publish actually about a year ago in Jackie in Practice in kids with long COVID, that they had increased AX and increased R5 minus R20 as well. These are obviously milder. They had milder acute COVID. I think some of them just continue to have it. They don't necessarily grow out of this small airway issue. So.
Thank you very much. First question is, as far as I understand, the spectral oscillometry is average values of the reactance and resistance, and intra-breath oscillometry is at the end of inspiration or expiration. The point is flow is 0, so it's not average value. Yeah, so it's more dynamic and FRC level measurement. But I hope Professor Dr. Hantos will explain following presentation, and maybe Dr. Chow can take over.
So we're gonna go up the levels, but I think like what we're seeing is that there is greater correlation of the functional disability with the intra-breath. And what the intra-breath does is that it actually tracks changes in lung mechanics during breathing. And so one of the things that Tadahisa showed earlier is that, you know, you saw those loops where he had actually colored it in red, is really telling us in terms of what the changes are during breathing. And so one of the theories I'm thinking about is that part of the reason for patients having the dyspnea, the sensation of dyspnea and the decrease in 6-minute walk distance is that even during tidal breathing, there are already changes in the lung mechanics. And so, which is not normal, because if you see I don't know if you're able to put that back up, but you know, if you look at the loops of the intra-breath loops during normal breathing, there is no change at all. Is that, you know, it's almost like an envelope, in terms of, so if you think about sort of where the arrow starts on the far right graph, so that is actually the resistance in expiration. So that arrow, that red arrow on the top is bringing expiration. And so you get down to, you know, to the other corresponding area where it's low as zero. That's the end. So you see that there's this huge change in the change of changes in lung mechanics. And I think that's actually what we're picking up in terms of the symptoms. You know, that's where the symptoms of, you know, of dyspnea and that sensation comes from. That's my theory. And I think we're seeing that in terms of the correlation analysis and the, you know, so it's more sensitive because with the spectral oscillometry, the measurements that we're getting is actually the mean of that entire breath. So I would be very, you know, I think this is a worthy of a point of discussion if other folks have thoughts.
Well, one question is where, you know, what is giving rise to those widening of the loop? Is it actually a mechanical effect? If so, where? The other thing, and I'm just going to throw this out there, is that, you know, changes in glottic aperture. Now, I'm going to speculate wildly here, but COVID is known to have neurotropic effects. Is there any chance it could be affecting glottic control?
There's a very good chance. In the lead study, we found differences in asthmatics where the inspiratory resistance was elevated, and in patients with COPD, as you might guess, it's on expiration. And it's just the nature of the changes in the anatomy, if you will, or the structure of the major airways that you're seeing differences. I always think about it like Mead-Wittenberger loops, which are pressure against flow and the slope is resistance. And inspiration and expiration are independent sorts of states. Now, in a normal person, it's very linear. There's not much effect, but in patients with disease, it isn't. And then the question is, what do we learn from it?
There's a very characteristic pattern of glottic breaking. And so I have to admit that I haven't looked at
so what
100 of those loops.
So in babies, for instance, use the vocal cord glottic breaking, keep their FRC up. Peter Macklem showed years ago that in normals, but in asthmatics as well, the beginning of expiration, resistance goes quite low, and then later in expiration, as you presumably flow limit on the flow volume, and the more
Right,
you get.
they're adults, so that's
These are all adults.
right.
Well, Zoli can correct me if I'm wrong, but I think with the glottic breaking on the one that's in the flow domain, resistance, you get this V-shaped character where it goes up on either end, but it comes back close together. So we're paradoxically measuring resistance under zero flow conditions, which is counterintuitive, but I guess it's the limit theorem as we approach it. Nonetheless, if we're doing it under zero flow, does that not take away any of the glottic aperture issues that you might have been invoking, saying that might be the explanation?
Not
Because
necessarily.
as long as the glottis is a little bit open, if there's no flow, does it really matter whether it's
But Ron, not necessarily, because if you are heading towards a particular zero flow condition, what's important is what's the resistance under those conditions. And if it's different at the two, then I think it will look different.
Okay.
But, you know, it may have nothing to do.
We may have some online help.
Yeah, sure. Let's go back to the gallery. Yeah, okay. Dr. Hantos.
Thank you. Just a short comment on this. So the intrabat tracking of resistance and reactance gives an opportunity to give some insight where the resistance increases take place. So if they are at the very periphery, which don't supply expiratory flow in expiratory flow limitation subjects, then in the central part of the airway system, there is no flow enough to drive into the upper airways into nonlinearity, even if there is glottic braking. Glottic braking is typically following the expiratory flow pattern, and it basically stops at the end of expiration. So sometimes there are very clear markers in the resistance versus flow and volume loops and reactance versus flow and volume loops, which help identify the locus of the airway constriction.
Yeah, and one further thing. I don't think it's ever been done since, but Peter Macklem with Raphael Delaca actually measured oscillometry and with the esophageal balloon directly measured flow limitation and found that these were COPD patients with fairly, I don't know, stage COPD, but it was fairly advanced. So for sure, you can measure the reactance loop. You can directly relate to flow limitation.
Yeah.
Whether that's flow limitation in these subjects, that's a different thing, but the loops are not that dissimilar.
I'll also tell you that over time, the dyspnea symptoms do seem to get better. We put them on an exercise program, and that seems to be helpful. It's not clear if it's the program or the time, but the oscillometry findings still seem to be there. So I'm not exactly sure why, but it seems that there is some disconnect between, these findings and the actual symptoms.
Can I ask?
Is being recorded.
Can I ask it, with the long COVID, is there any evidence sort of at the end or, while you're well into the long COVID, is there any evidence of active inflammation still going on? Or do you just have, what I'm asking is it an active inflammatory process or is it sort of a fixed remodeling? Do you have any sense of that?
I think it's the latter.
Yeah.
Actually, if you do look at inflammation like cytokines in the blood, at least for kids, this is not necessarily true for adults, you actually have less inflammation. So less, especially Th1 inflammation. That doesn't tell you exactly what's happening in the lungs, but certainly, FeNO is normal. So I don't think this is inflammatory. So.
Well, and again, this is speculating wildly, but being shown in animals that you can have fibrotic deposition fairly soon after an allergic inflammation, but it's not totally permanent. It will go away eventually. So, we think of laying down of collagen as being permanent remodeling, but it could be transient remodeling. So, but with a long time course, maybe that's what's
just it doesn't cross-link, it'll go.
Yeah, right. It may not.
Yeah, Julian Allen.
Hi. Yeah, I just wanted to point out, in addition to the widening of the reactance loop that Jason was talking about between inspiration and expiration, one thing that wasn't shown in that diagram from Zoli's review article was in Chung-Wei Chow's work, there was also a marked decrease in the end inspiratory reactance. In other words, a more negative reactance at end of inspiration. That you don't really see that in Zoli's picture, but to me, that's another important aspect of intra-breath work, is that change between inspiration and expiration in the stiffness, which I interpreted as being increased stiffness on end inspiration.
Thank you for the shout out. But I mean, in our IPF population, it is EXI that is the clear marker in terms of severity of restriction and prognosis.
Okay, thank you very much.
Thank you very much.
So Dr. Jeff Maxims finishing off with a similar discussion on long COVID in a milder population.
You gonna pull it up?
For sure, yeah.
It may be on the far left of the screen. Yeah, I think they are. Far left, just above the Zoom, and is that Jeff?
That's me.
Ah, okay, great.
GM.
GM. Okay, so let's go back here. Oh no, it's not open yet. Sorry.
Right.
Okay.
You
Now I
gotta
just
quickly
have to
change
share.
the slide because I
That's
got the number wrong.
Okay, so you want to go back to edit mode? Oh,
Is this right? Yeah.
sorry, your last year's a seven.
My apologies.
We wouldn't have noticed to be had. It was a good time last night when you made the slides.
That's it. So disclosures, yes, I'm a co-founder of Thoracis and sometimes advisor. When they want. Anyway. This is gonna be two parts because I'm gonna talk about quality control at the end.
Jeff, stay in the camera, please. Oh, you know what? We might actually have to put it up because you're so tall.
Where is the camera?
I know.
This thing?
No,
Oh, you do it.
That's okay.
Between
Okay.
this board and this board.
I have to stay here. I got it. Okay.
That's good.
So, I'm glad that, I was preceded by, your group, on long COVID. And, so I don't have to spend as much time describing it. But this population that I'm going to describe is quite different. We're getting them later after their original COVID infection and they're milder. They weren't admitted to the hospital. Okay, so the long COVID Canadian study was originally to identify the prevalence in long COVID after infection in the general population, so including all comers, and identify risk factors contributing to long COVID if we could, determine the spectrum of severity. Number 4 is what I'm going to talk about, which is determine the physiologic objective characteristics of respiratory long COVID. Basically look at the patient's pulmonary function tests, lung volumes, and oscillometry. So we included patients who would, or subjects that had previously tested positive for SARS-CoV-2 or exhibited symptoms and diagnosed with long COVID. And we also included subjects who were tested after testing greater than 6 months. That looks confusing, but less than 1 year after the most recent positive test for SARS-CoV-2. So far, there's 466 patients. The study is largely complete, so there won't be that many more. And there's a recruitment bias because it was a long COVID study and we're recruiting patients for that. And so patients came, 55% of them had diagnosis or symptoms of long COVID, and 42%, although they had acute COVID, didn't have long COVID. And 65% of those with long COVID had what we call respiratory long COVID. That means they had respiratory symptoms, but some of them had the other symptoms like fatigue, brain fog, tiredness, that sort of thing. So we collected a lot of information: ethnicity, living alone or with others, education, employment status, income, vaccination status, smoking, vaping. We have comorbidities, so the respiratory diseases that they may have, and a fair number of these had some. They did a number of the questionnaires, so a couple of those are important for the symptoms that we extracted. And some of them are like health anxiety, which is another hypothesis about some of the people with long COVID. So we had 466 subjects, 166 of them, 65% had respiratory long COVID, and 35% had non-respiratory but diagnosed with long COVID, and the rest had no long COVID. Looking at a little bit in detail in terms of their respiratory symptoms. So if they had any symptoms, there was half of them had any symptoms. Respiratory symptoms was about 166, and non-respiratory symptoms along for that amount. And this is the kind of symptoms that we collected. I don't have time to go through the exact details there, but there's some differences. And I said the study is largely complete, and we're transferring the data from all the sites. So actually, wasn't able to analyze anything except the Halifax data so far. And so the Halifax data, we have 33 non-long COVID patients and 55 respiratory COVID patients. And the demographics are there, male, female splits. We have more females with respiratory COVID and with non-long COVID, so more females were recruited into this study than males. But the age and height and weights are largely similar. So the first results we're showing here are that, and these are the p-values. This is just t-tests, and then there's t-tests for multiple comparisons, corrected p-values there. And you can see that just this extraction of the data so far, R5 was not significantly different in this group. And this is not intra-breath intervals. This is R5 averaged over expiratory minus R5 averaged during inspiratory. But we do have the 10 Hertz data. I'm not showing that yet. However, the X5 was significantly different between the respiratory COVID and the non-lung COVID group. R519 didn't reach significance either. So in milder subjects, it's X5 that seems to be the dominant distinguishing feature. And I'm not presenting the pulmonary function data that's being analyzed in Alberta, but I've been told there's nothing. So nothing happens. Yeah. So that's it for COVID. I'll return to a little bit at the end. But I want to discuss a little bit about quality control because I presented previously at this conference on quality control, but it was just spectral oscillometry. Now we're moving into the loop analysis. Okay, so, and there's been discussion yesterday, so I adjusted some of my slides. This is an adjusted slide based on what we were talking about yesterday. So the technical standard, that's the King et al. Paper here, in ERJ 2019 has this statement. It is recommended that 3 replicates used to derive indices should have a coefficient of variation of resistance at the lowest oscillation frequency, which is R5 typically, of less than 10% in adults and less than 15% in children, although there are currently no published data to support these cutoffs. So that's a very strong statement. It's not a strong statement. Right? These were experts that sat around a table and chose those numbers, right? Based on their experience with the data. The worst way to do it, but better than nothing, maybe. Then later, there's this sentence, like one or two more sentences. The use of COV and its specified threshold value to select replicates should be declared in the report or methods of publications. That, at the time, was thought to be, we suggest less than 10% or less than 15%, but make sure whatever you suggest, put your number in, which means if you have good reason to choose a different value, right? And I think that wasn't clear in the manuscript. I'm saying it now, right? Now, Zoltan published just recently quality control in respiratory oscillometry, reproducibility measures ignoring reactants. And so, in fact, I'll just go to the next slide. This is the data he showed that there's a lot of variation in reactants of a similar magnitude to the variation in resistance at 5 Hertz, right? And this is not contributing. But similar to the discussion we had, why not choose Z to report to the greater masses of respirologists and clinicians out there? We could use Z to actually quantify the quality control, because Z is actually the magnitude of your signal, right? You're getting some resistance, you're getting reactance. Why we decided to focus on resistance is probably historical momentum. When oscillometry was first developed, resistance was the thing people wanted. And even some papers would ignore the reactance data in their discussions.
They're using reference data that ignores reactance. So resistance has had this lens of focus on it, whereas reactance has not. Now reactance is known to be very sensitive to peripheral airway disease, and it should be included in the quality control metric, in my opinion. This graph basically shows that impedance, this is the length of the line here, is the size of your signal for, well, this one, the blue one, which is actually, if I rotate this, is longer than the, what do you call it? The vector of, there's a better mathematical expression. Anyway, the portion of the resistance, there's a portion of the magnitude of impedance expressed by this length only on resistance, and by this length only on reactance. This length is the longer length. So you get the strength of the signal as the R squared, because this is Pythagoras' theorem, plus X squared equals Z squared. Anyway, so I strongly support that recommendation from Zoltan Hantosh. But I'm going to raise the question. So if you were moving in that direction, there's been no publication that actually establishes a cutoff or a threshold on Z yet, but we should move in that direction. But what about quality control for the interbreath analysis? If you want to, can we achieve, looking at this sentence down here, can we achieve reproducibility, a metric, or even our, whatever we decide, that's reproducible, I can trust that measure, similar to the coefficient of variation for spectral oscillometry. And it's actually a difficult thought, because coefficient of variation from 3 repeated measurements of resistance is just 3 numbers. You do the mean divided by this, the standard deviation divided by the mean, right? And if the standard deviation is 0, you get a very low COV, right? 0 is perfect. It's absolutely reproducible. And 10% is pretty good. And it gets worse. But 10%, 15%, 20%, there's still somewhat reproducible. But how do you do it when you have loops, right? So anyway, so we've thought about this a little bit. And you want to have loops that are following each other, overlapping with each other. A loop with an artifact would be some zingy loop that goes zinging way off into space, comes back. Maybe this one here, and this particular trace is something, I don't know what that one's doing, and most of the loops are down here. So by eye, you might do it. Someone might actually pick this. This is a kind of technique that's actually been done by hand, right? These loops look all reproducible, so I might include all of them, right, rather than eliminating.
But Jeff, can I interrupt?
Yeah.
What if your glottis is spazzing out and that's the signal? Then you want to have a look at the variability of the loops. That's what you.
Well, yeah. I mean, if that is happening, if your variability is reproducible. Everywhere.
But even if it isn't, it could be lack of glottic control could give variable loops, and that could be actually the physiological signal here.
Yeah, yeah. And well, we have that problem with the spectral method as well. Right? So if our current standard has the thing that we want 3 reproducible loops, and the one that's not reproducible is not measured, not included in our clinical data, right? An expert respirologist looking at that one might be real. Right? So we should have that proviso that you should pay attention to the outliers. They may not be artifacts. Okay, but we are only learning so far as to what an artifactual data looks like versus real. Anyway, so we're starting somewhere rather than not doing anything. So what we thought about was, okay, well, we'll start from a similar space. If that's the current technical standard for spectral, then we want 3 reproducible loops at least, and then you've got enough data. Right? So we developed a cost function based on Z. So it's based on the sum of square distances between points between the loops that are going flow Z on the y-axis, flow on the x-axis, and you have 2 loops. Like if there's this loop and that loop, there's a huge distance between them. So that COV equivalent would be an enormous number, right? But if the loops are overlapping, it should be 0. That's the idea. So it's distances between the points on each loop, right? We had to do some things because not every breath's the same length. So you actually need to figure out what are the closest points. So you have to do that sort of thing. I'm not going to go into the mathematical details of that. And I threw this problem at a bunch of undergrads. So in engineering, you have to do capstone projects. So these are year-long projects, and it's design. So you have to design something. So I thought, well, why don't they design a tool to do this loop analysis? It's a bit of science, and they also produce this an app. This is basically the front screen of an app that, should please. I said, well, maybe we want to show this to the company one day. So they got the idea of putting the logo to make the boss happy. Anyway, so that's what's up there. They got that off the internet and stuck that there. But this would be the kind of display. So this is a number of loops. 3 of them are in bold. And those are the 3 that are chosen that the algorithm basically gets the cost function and determines the distance between all the loops, all the permutations you can possibly have to see if there's 3 loops with what would be the cost function, the distance between the best 3, the next best 3, and the next best 3. And it doesn't really say, but this is the volume cost function and this is the flow cost function. And we haven't quite figured out exactly how to normalize them. It's basically that you can't divide by you could divide by the mean, but you could divide by the peak to peak along flow and that sort of thing. So that's what this one is. It's peak to peak. Anyway, so as the difference between the size of the loop from peak to peak, this loop is off by 1.8%, 1.87, but not, I'm just doing the X direction. It's actually in XY.
So Jeff, can I say, did you sort of say, okay, I'm going to say one particular loop and then do all the permutation of all the other loops and say, okay, these three work together, and then
yeah, so you end up with, if you have 9 loops, many loops, 9 factorial, whatever it is, and then it finds the 3 that are closest have this cost function, and so that the clinician learns a little bit, it does the top 3. And when the clinician is, or whoever is using this, they click first, they see the first 3 light up, and then they see the second. So they can see how sensitive it is to which 3 you're choosing. And maybe this won't be the way that the ultimate measure is implemented, but this is a starting point. It could be that here's the 3, and then what are the distances the next one? And if it's close enough, include it, something like that. There are ideas. These are the names of the capstone undergrads who created this application. And so here's, unfortunately, I don't have any from COVID, but we have some lung transplant patients. So this is X10 versus volume and X10 and R10. So the resistance on the top and X on the bottom, same axes, and versus flow. And you can see the opening of the loop here for resistance, not reactance. And there's differences, like there looks some wonky loops here that got eliminated, but this algorithm basically picked 3 that are supposed to be reproducible, and so they kind of look like that.
Are the dotted lines the wonkiness?
The dotted lines are the other ones in the recording. So there's not actually many breaths, because this is a 30-second recording, right? And so there's maybe 8 breaths, something like that. So just looking for feedback on that. Actually, I don't think Thomas has seen this. So feedback from him. And where do we go from here? Because it's a starting point for working in the same space that we're familiar with. 3 good breaths. It's reproducible if 3 are below a threshold. We don't know what the threshold will be and if this is the right metric. But this is a trial of what that could be. So, but at least a cost function can be developed, analogous for the coefficient of variation for spectral standard oscillometry. And going back to the earlier part of my talk, preliminary data indicate that subjects with respiratory symptoms may have lower X5 and X5X in relation to subjects post-acute COVID, so long COVID. So they have something that's different in their lung functions that's lasting, if I take the middle ground, a year after their last COVID test. 6 months to either side of that. And although I don't have the data here, when we looked at, tried to correct for patients who had lung disease, that actually, the relationship disappeared with patients without lung disease. So if we take people, if we just looked at patients who didn't have lung disease, we didn't get a relationship anymore. So the critical person might say, well, you developed a great tool that detects lung disease, right? But it also might be, because that's basically what happened, but really what it means is that patients who have COVID and lung disease get this, right? Patients without lung disease may not have that relationship, and they got COVID, right? So, but they might have another form of COVID. But I don't have the statistics on that, so that's hopefully what we'll find when we expand to the multi-sites. The problem with that was it reduced my N value quite a bit, so I can't really say anything other than I'm hoping for that, which is not really good, but that's what I'm hoping for. Thank you very much.
Questions from the room?
To what extent do you expect inter-site variation in, you're talking about worrying about just one patient and lots of variation. What about across sites and operators in this study? How are you going to address that?
Yeah, well, in training is an important aspect. So, every site was trained the same way, right? But there's still different people. So there's going to be a dependence on the operator to some extent, but it's, Chung Wei published on this already. Okay. And training is very important, and it reduces the variability between sites, but the variability is still smaller than what you hope the disease is. Right?
Yeah, but you should probably statistically, in such a large data set, there are ways to.
Yeah, we could probably look at that.
To do that, I had just one other question as a novice in oscillometry. When we did our study with veterans, we did whole breath values. And I noticed a lot of emerging papers, talks of doing expiratory minus inspiratory or vice versa.
Yeah.
What should we be doing clinically?
You're asking an engineer.
Okay.
I'll jump in on that. What you can do, depending on the device, continue gathering spectral, because what we're working on, both Chung Wei and I, in conjunction with Tolly, who's really the brain behind this concept, is we can take those spectral oscillograms that you're generating and do intra-breath analysis at specific frequencies. And you happen to have 11, which is close enough to the 10, that although loops may not be as pretty, statistically, the data you generate is essentially the same. And we've actually published that already, so that's new, not news. What's news is that it means that we can now access existing data sets and retrospect and extract intra-breath from them, depending on which device you use.
Yeah, but there's physiological reasons why the intra-breath has potentially more sensitivity than the spectral. And it's simply because if you're doing averaging through the breath and inspiratory measures are normal, right, but expiratory are different,
right,
then it's better to just look at the expiratory. And then again, instead of just averaging the expiratory, if you have nice reproducible data, then you can trust the peaks, which is at end expiration or end inspiration.
Yeah,
and look at the differences. And so the levels of increased sensitivity theoretically are getting better and better, as long as we have good signal to noise and reproducibility.
Like in the lead study, they had considered expiratory breaths as well abnormal. They did a sensitivity analysis.
Yeah, we showed that the expiratory was abnormal in patients with COPD, and inspiratory was abnormal in patients with asthma. And we were able to distinguish the two groups based on that.
Yes.
Yeah. And there's some good reasons to believe that that's the case. So I see this as analogous to flow volume loops.
Yeah.
I think that people who interpret lung function without looking at all the loops, you miss things, particularly vocal cord dysfunction. And then as Julian pointed out, and we talked about yesterday, is the variation of the loops related to some aspect of understanding the disease. So as an example, if you've got a lot of bubbles and mucus running around in the lung, you might expect to see a lot more increase in the looping and a lot more variation. And that maybe that's the signal it's
Yeah. To your long COVID stuff, and maybe you can answer this, Jeff, or maybe Chung-Wai and Ron, who look at impedance in patients a lot, might be able to answer this. You had a significant increase in the magnitude of X5, but it didn't strike me as a particularly impressive increase physiologically. So if you study patients with asthma or any other kind of lung disease symptoms, would you expect an increase in X5 of that level to be associated with noticeable symptoms? In other words, are the symptoms that the long COVID people experiencing, are they reasonably associated with the change in lung function, or might you expect something else?
Well, I wonder if it's more deconditioning driving their symptoms. They have a decreased level of activity, because if the reactance is dropping from like -0.5, -1 to -2, -2.5, I'm not sure that they're really going to perceive it, because you're right. I see much more profound drops when I see my patients that are symptomatic. So I agree with your criticism there, but I don't know what Chung.
Well, it was a question.
Yeah, and I think there's a component of that, but I think the other component is actually pulmonary vascular disease.
What's our
Pulmonary vascular disease, and what was really unfortunate is that during that period of time, there was just no ability for us to get full disease and diffusing capacity or imaging on any of this cohort. But I think some of the other studies have, I mean, there's clearly pulmonary vascular disease, which I think responsible to.
There's also a lot of cardiomyopathy that's not always well appreciated.
And
I've also heard people invoke, I mentioned it before, neurotropic effects. But it seems to me, once you do that, you can, I mean, that's a bin you can explain anything by, but not actually explaining it. Invoking magic, so who knows?
And just there's a handful of experience, including us, who have transplanted patients post-COVID. And most of these folks are young, previously healthy in as much as we can tell, but their outcomes are not good. So it's very different than other, healthy populations who get ARDS and then get transplanted right away. That's why if you remember, there was like all this excitement in the news about transplant, and then you don't do anything thereafter.
But with the transplanted lung, you could potentially understand whether there's persistent infection going on at a very low level. That have been speculated about at all?
Well, everybody, I think infection is a big issue post-transplant.
Right.
And the infection burden is not so different. I think what's different about this cohort is that if you look at it.
But if it persisted?
I
mean, this has been bandied about as a theory that causes asthma, particularly in kids, is that you get a viral infection that then somehow magically changes the lung, make it much more hyper-responsive through some presumed genetic issue. So I'm wondering if it's something similar.
I don't have any objective data.
No, I'm guessing. Wild.
Well, maybe take the last question.
Yeah, exactly. Yeah.
Little over time.
That's right. So back to you, Dr. Rabinovich.
Yeah, so just a couple of comments. First of all, the presentation is just so different than asthma. These patients come in, they are, complaining of shortness of breath and sometimes often at rest, which is, not typical of asthma, of course. And, you know, when we think of that, we think about vocal cord dysfunction, we think about, you know, upper airway issues. So when we do like real-time laryngoscopy, a fair amount of them do have closing of the vocal cords on inspiration. And then some of them have this kind of, I don't know, wastebasket type of thing called breathing dysfunction. They hyperventilate as well. So I think that there's a bunch of different reasons for their dyspnea and probably not just one. And I think being able to tease that out is interesting. So I'm interested in this idea of lack of glottic control actually being the reason for, you know, the higher reactants, and whether you could in some way differentiate, let's say by giving them albuterol, because some of them do respond, their AX actually does, you know, decrease quite a bit, not up to the 80%, but it does decrease with albuterol. So I don't know if there's a way to.
Study, we did include albuterol, so there's pre and post data. Just haven't analyzed that yet. Be able to look at that.
Okay, thank you very much.
Yeah, thank you.
First half. I'll just ask everybody to come back in 15 minutes. We have a coffee break.
I'm pleased to introduce Zoltan Hantosh. Take it away.
Thank you very much, Jeff. Am I audible?
Yes.
Okay, thank you. Thank you, Jeff. At the expense of my talk time, I congratulate to your talk, and I'm very happy to have this positive reaction on the unread and uncited paper of mine. Thank you very much, and also pointing to the necessity of the green grass of the impedance plane, where probably we make again friends with Charlie, and I'm already less grumpy than yesterday. Thank you very much. So let me try to share my screen, which is this is the bad screen. What happened?
Let's read his email.
Yeah.
Yeah.
One of your other screens.
Yes. Sorry for that.
Ah, good, he's got people.
Yes. Your manuscript didn't work.
Is it possible to have the presentation view?
No, he's getting help now.
Okay. It's getting better with the help of Shari.
Excellent.
Perfect.
Good. Thank you very much. So, first of all, I show you some disclosures here. And more importantly, I want to show you this motto from Janet, which is probably the best summary of the adventures of infant oscillometry or infant lung function in general, because it is probably not oscillometry specific problems which I want to raise. So the main issues is the higher specifications from equipment and the meticulous attention to detail. So this is very important. In this study, we want to address the effect of how the face mask is held against the infant's face, how the head is supported, how the mask cuff inflation affects the impedance data which we receive, whether the open mouth and closed mouth conditions differ, because we expect that with open mouth or mouth breathing, the impedance would be probably half as much as we have with mouth closed, that is through the nasal pathway. And there are issues which should be addressed as the method of calibration and the computation of impedance. So in this Indianapolis study, 26 preterm, born preterm infants were studied and measured twice at the age of 5 to 6 months and at 12 months. So this we call a visit 1 and visit 2. The oscillometry device was a tremor flow device in development called N100 by the Thoracis company. And this used 7 to 41 hertz frequency range. I mentioned that several 16 hertz interval measurements have been made in the context of this study, but I'm not going to report unless you definitely ask me to do that. The infants were measured in the supine position. They were sedated. In one study, the head and mask support was accomplished by one hand of the operator, and the measurements were repeated or repeated with the two hands version. But in both situations, the operator concentrated on the baby and was not dealing with computers and other things. Minimum of 3 segments or 8 seconds, not 7, of regular breathing were recorded, but not unlike the recommendations for the oscillometry. The face mask was not removed in between because otherwise the babies would have been provoked and with the facial nerves stimulation. So that is totally inappropriate to remove. We are happy if the child is sleeping nice and we can make recordings in a one long recording or in segments. This is a schematic figure from borrowed by Farah Al-Huda, who did her master's thesis on this with this device in development. So it means that the original base unit, which is continued directly with a bacterial filter and mouthpiece in adults and larger children, is substituted by a corrugated tube leading to a flow head, starting with a pneumotech. That is, the flow is measured locally here. The pressure is also measured here. There is a pathway to breathe through for the infant, but otherwise, a bacterial viral filter is interposed between the measurement head and the face mask. And also because of this relatively large dead space, this port of the filter is used to introduce a bias flow of approximately 2 to 5 liters per min, depending on the infant's age, to flush out the equipment. So the setup is basically a measurement head, which is continued with a short tube, a bacterial filter, another short tube connecting the filter and the face mask. And normally, if we have our own wave tube impedance measurement setup for infants, we account for these in series components of the equipment part, and also the parallel compartments, which are basically gas compressibility before the measurement point, the pressure measurement point, and the half of the filter. Another compressible pathway is the other half and the face mask gas, but most importantly, the face mask is also a viscoelastic unit, which, because of the soft rim and the opposing soft tissues of the infant face, create a very, very significant viscoelastic shunt effect, which we measure in parallel after removing the former components in parallel with the real respiratory impedance. So we are specifically addressing in this study the face mask shunt impedance, which is not taken into account in the original N100 software. We addressed the impedance data, which are reported by the Tremorflow N100. We use impedance estimate by a custom-made software which process the pressure and flow exported signals from the Tremorflow, and also the face mask corrected impedance data. And these impedance measures can be single frequency values of resistance and reactance, resonance frequency, reactance area, the frequency-dependent resistance. So the usual outcome measures which are routinely reported in oscillometry. In addition, we use model fitting with a very simple model, taking basically the average of the real part of impedance, that is resistance, and the imaginary part of the reactance part of the impedance is fitted by a compliance inertance model to eliminate to separate these two parameters which are influential at the lower and the upper part of the frequency domain. This is an example of just pulling all the reactance, resistance and reactance data with two-hand support and one-hand support at visit 2, that is the 5 to 6 months old children. And by eye, you can see probably two impressive things. One is that the two spectra are basically similar, and actually the statistical comparison did not reveal any significant difference between the two-hand support and one-hand support. The other thing which is very obvious, that in these raw recordings at the younger age, basically resonance frequency is not reached between the 7 to 41 hertz range, which was a little bit surprising, but we will return to that later. We have lower resistance values and higher reactance values in the second visit measurements, and we have now quite definite, although not exclusively measurable resonance frequency in this range. Again, the one-hand support and the two-hand support don't show any difference, and there's no significant statistical interaction between the visit 1 and visit 2 data in terms of impedance data and age. Well, if we process these data, these signals, with a custom-made software, with no correction for the face mask, with a relatively prudent correction, and probably a more likely correction. So this is 1 to 100 to 67% range differing. So this is a range of the stiffness of the face mask touch on the infant face. So in the first correction makes some difference in the resistance values, but from that point on, and including the tremor flow reported resistance data, basically there is no difference between these levels. In terms of compliance, which we derive from the model fitting of the reactance data, in both the custom-made software evaluation and the tremor flow reported data evaluation, we have an increase in compliance with more compliant mask impedance comparison. So if you take into account a relatively rigid face mask, the compliance goes on, and further elevation is observed if we assume a very soft touch. In fact, we can make some bench tests, pushing the face mask against a soft body surface like cheeks or lower arm and things like that, but it's never exactly reproduces the in situ shunt effect. So this is an approximation. AX is very sensitive to this. So if we don't have any compensation for the face mask, we have awfully high AX values, which are falling very fast with the softness of the shunt impedance in both the custom-made software and the tremor flow reported data. Very apparently and very surprisingly, if we take the pressure flow signals and compute impedance with a custom-made software, we don't have the same values as the tremor flow reported impedance data. So without any correction, we don't have resonant frequency in any of these hits, putting the correction gradually more and more occasions of resonance frequency are detected, but eventually the whole number, which was 24 in this particular comparison, is reached only when the face mask support is compensated for. And this is what I would like to stress here, that the original pressure and flow data, there is no frequency dependence of reactance. With the correction, we have some. So this is a basic contradiction between the reported pressure and flow data and what we process from that and what the tremor flow software processed. The other step, which is less disappointing, was the mask cuff volume. So we can see that the R7 data are basically independent of the cuff volume from between the 10 milliliter and 25 milliliter range. Some decreasing tendency might be seen at the 41 hertz resistance, but the change is statistically not significant. The same results for the X7 and X41, and very convincingly, the C values are absolutely independent of the cuff volume. AX is slightly increasing, but still not reaching statistical significance for this trend. So this is very reassuring that it is not very critical what a cuff volume is inflated. in the cuff, because probably the harder or the cuff itself, the less pressure is exerted against the baby's face. That's my impression. The other comparison is based on a fewer number of recordings, and now we pool the 5, 6 months and the 12 months data, and just compare the closed mouth measurements and the open mouth measurements, and basically we don't see much difference. So if the mouth were opened, but the nasal breathing was maintained, this means that the closed down oral compartment is made in parallel with the face mask compartment. It makes a little difference, but not much. If it were real oral breathing, we would expect the large force in resistance, because the nasal pathway typically accounts for at least 50% of total respiratory resistance. And the same picture is received if we accomplish one correction. Resonance frequency goes down, but this is the same with closed and open. So open mouth, if is there, is not automatically associated with oral breathing. Overall, the raw data, visit one data are shown here, the individual lines are compared with 6-month-old Bangladeshi infants from a cohort study, which gives fairly good agreement at low frequencies with reactants. But resistance in this Bangladeshi cohort is much higher. It's probably the nasal pathway problems in that population. But these measurements, which were made with the wave tube technique, display a 30 few hertz resonance frequency, which is not reached in the Indianapolis population. The other comparison is from our neonatal study, healthy term infants measured in the first 3 days after birth, and good agreement is seen between the resistance values, but the reactance values are totally different from what we see here. In phages, there was a comparison of the wave tube measurements and the N100 measurements in a very limited number of babies. And R7 values were systematically higher according to the Bland-Altman comparison, and some trend, but it's very, very careful, just 4 points, of course. X7 does have a difference, but not that systematic. And at the highest frequency, at 37 hertz, the overestimation or underestimation of resistance was clearly seen at resistance, and the same picture is seen here. So this is very, very small sample to compare, but this illuminates that is something very, very inappropriate with the calibration of this device. So our conclusions are just confirm that we have very high impedance values in infants, which, even if you speak about average impedance, interbreath changes might be in excess of 100% of the average. And if this 100% is misestimated, it bounces back to the problem of the mean value reliability. So the calibration and the validation is absolutely imperative in the measurements of high ZR's values. And the other take-home message of this comparative study was that we can't copy the protocol and or minute it to the size of the infants just by copying the recommendations. This is a different situation. Supine, sleeping, and nasally breathing kids, and it's as we often encounter, it's very difficult to reach these sleep stage without sedation and with limited time for the study. So thank you for your attention. Oh, I didn't want to show this introvert stuff, so I thank you for your attention. And stop sharing.
Thank you very much, Zoli. Let's see. Don't see any questions yet from the chat. So are there questions? Jason, go ahead.
Yeah, hi, Sultan. Very interesting, of course. One thing I noticed is that the data that you showed, you had a fair spread in the resistance and elastinces. Like, I think resistance varied by about a factor of 3 of the population you had, and there was a pretty big spread in reactance as well. What do you attribute that variation to within the population that you looked at? Were they different sized kids, or was it different nasal cavity size? Where do you think that spread comes from?
So if you look at the similar age measurements, so visit 1 and visit 2 are not combined, the scatter is still there. So since these impedance data incorporate the huge nasal pathway resistance, which is unstable. So the nasal patency is a critical thing. And I don't know if there was any irritation or something like that. In the Bangladeshi study, we did notice that the nasal patency was critical. But even with normal populations, like the healthy term neonates, we had a great scatter. And also this temporal variability was very huge. So from day 1 to day 2 and day 2 to day 3, very, very different measurements were attained. As for the reactance data, it can probably more reflecting the FRC level in these unstable and expiratory conditions in neonates and young infants. So the variability in reactance is probably mostly attributed to this pulmonary working point instability. But the resistance is probably more linked to the upper airway problems.
Yeah, thanks. That makes sense.
Okay. Julian, I see you have a question.
Yeah, thank you. Zoli, that was a fantastic talk, really important work and very difficult to do. I have 2 questions, Zoli. One is, do you have a sense of what the coefficient of variation is in your measurements, the intra-subject coefficient of variation and what we're willing to accept? And the second is, so how do you think, what's your vision of how we then do longitudinal studies from the infant through the child when we're moving from the face mask to no face mask? Do you have any thoughts about how we can actually do longitudinal studies then?
It's a very good question. The first issue which you raised is I can't tell you the CV by heart, but these were inter-individually very reproducible. And it's just because the very meticulous and very professional laboratory personnel. So the one-hand, two-hand measurements were highly, they were different in time. So one set was collected 5 measurements with one-hand support, 5 measurements with two-hand support, and things like that. And the reproducibility was amazing. I just guess that the sedation might have improved this reproducibility.
Yeah.
The other question is very fascinating. So when we switch from the supine position to the sitting and from the nasal to the oral breathing, this is basically a different side of the river. So they are hardly comparable.
Yeah.
There is a jump in the oscillometry outcome measures anyway. So we encountered this problem with the South African follow-up study, and now from the 3-year-old children population is starting to feel the 11-year-old. Everything is fine, but the gap within the 0 to 2 years and from 2 to 3 is there. So I can't.
Yeah.
Probably in the forced spirometry studies, there's a gap because the FEV05 and the FEV1 at some age has to take over.
Yeah. Thank you. Fascinating talk.
Thank you for that question. I think in the interest of, quick one there, Charlie?
Yeah, it's quick. So I gather the problem with the face masks is its compliance. Is that correct?
Yes.
So couldn't you
Yes.
use a face mask that's very stiff and avoid them?
Well, actually, we were fiddling with a different face mask in the infant studies. But one issue is we have to make it absolutely leak-free.
Yeah.
So we have to follow the different facial geometry and anatomy. And this is sort of optimization, how soft the rim of the mask should be. So first of all, eliminate any leaks, because otherwise the impedance will be crap and unreliable.
Yep.
And reactance inversion might take place,
Yeah.
which we sometimes encounter with some commercial devices. And this is just one part. The opposing tissues are another problem. So they together have a sort of compliance which is far higher than the internal gas compartment compressibility, which normally the first gas to address, but it is not true. So the gas compressibility is nil in comparison with the soft surfaces joining and oscillating. So this is a very difficult question and very, very, very hard to standardize.
Yeah, I would agree. Very challenging group to study. So I think thank you very much, Sultan. I think we'll move on to the next one. So we'll bring Yassine Laroud from the University of Toronto. Okay, all the way. And the impact of multi-frequency and sparse frequency oscillometry on derivation of AX values.
Thank you.
Right. Okay, we need to find your talk.
Sparse.
So This one?
No, second.
Based on first. Do this. Is the screen shared? I don't believe it is.
So I think that's what they say sparse. Yeah.
Ah,
Share.
yeah, the rest of it's only three frequencies.
Yeah, right.
Oh, yes.
We'll find out.
Yeah, but yes or no, because in the
Yes.
Excel data sets, it does have.
Thank you very much.
But I don't know if I think they're truncated.
Hello, everyone. As I've been kindly introduced, thank you very much. My name is Yassine Marouf. I'm a student biostatistician underneath Dr. Chow at the University of Toronto. And today I'll be mainly presenting some research that I've done underneath her on estimating resonance frequency and AX whenever we have different ranges of frequencies or even sparse data. So to give a little bit of background, as everyone mostly knows, the common measures that we use in clinics for reactance curves is usually the resonance frequency, X5, and of course the area under the curve in between them, the AX value. Moreover, we commonly have it associated with different lung physiologies and has been used in a pretty robust manner. The problem is that some patients some patients with abnormal lung physiologies tend to have higher than average resonance frequencies at above 20 hertz, which can be problematic since there are some more recent commercial oscillometry devices that are stopping at 20 hertz for the resonance frequency. So we end up having a problem where some of the patients that might have lung physiologies that we could explain with resonance frequency and AX for over 20 hertz that cannot actually be explained using those machines. So the predominant research questions that I was looking at was, what is the impact of resonance frequency and AX when we try to estimate it using this shorter frequency range from 5 to 19 hertz? And the second one is, can we find a different metric that would also be somewhat robust to these abnormal or normal lung physiology differences? So to give a little bit of background on the methods and the data. So thankfully, thanks to Dr. Chow and her research, we have a large range of oscillometry data. We have, at least in this research, we have over 560 patients with varying lung physiologies from normal to abnormal with resonance frequencies from 6 hertz all the way up to 36 hertz. Keep in mind, though, this is for the adult population. We're not talking about children here in this case. And as for the calculation for the resonance frequencies, what we used were the PIML models like Andy used and Jordan, I think, also used in the previous resistance compliance inertance model. And what we did is that we've specifically estimated those using only 5 to 19 hertz data. So if we have, the first thing we did was for 5 points, so 5, 11, 13, 17, 19. And then the second method was only 3 points, 5, 11, and 19. And if there were some resonance frequencies that were estimated to be over 40 hertz, we brought them down to 40 hertz because it's relatively improbable that an adult would have any resonance frequency above 40 hertz. As for the area of reactance, what we did was something similar. We used those estimated resonance frequencies, and there were mainly two cases. One, if the resonance frequency was underneath 19 hertz, we would calculate the area under the curve like usual, right? However, if the resonance frequency was over 19 hertz, because of the fact that we don't have the data about it anymore, what we did was we estimated a triangle from 19 hertz to that estimated resonance frequency and added that on to the area under the curve from 5 to 19 hertz. So what we see in these two figures here is a more clear visualization of a difference between a 5-point and a 3-point curve. Whereas on the top, we have a 5-point, 5, 11, 13, 17, 19. And on the bottom, we'd have only 5, 11, and 19. And what this red triangle signifies is what it would look like if we were to extrapolate what's left of the area between 19 hertz and the resonance frequency. So to give a little bit of results, first of all, for the resonance frequency, what we saw is that there was a significant difference between the measured resonance frequency from the tremolo flow, which I forgot to mention, but it was from the tremolo flow C100, and the estimated resonance frequencies using the truncated ranges. So regardless of whether we use 5 point or 3 points, there was an issue with the estimation. when we looked deeper into it by breaking it down, stratifying between patients under 20 hertz and patients over 20 hertz, what we found was that the estimates were somewhat robust. There was no significant difference for patients under 20 hertz in terms of resonance frequency. However, most of the inaccuracies came from whenever we have to extrapolate the resonance frequency for those truncated hertz. So anytime there was patients who had a resonance frequency that was measured at above 20 hertz, we usually found that there was also overestimation in terms of their prediction.
Can I ask clarification?
Yes.
You say extrapolate to the.
Yes.
Did you extrapolate or interpolate?
It would be extrapolate because the thing is,
Based on how? How do you extrapolate?
Yeah, exactly.
so
I didn't design the triangle.
I guess the way I'm using extrapolate was more in terms of the graph itself, not as much in terms of the equations that we use. So I guess it would be interpolation whenever we talk about the model calculations itself. However, the proper word would probably be trying to predict. Because
Right, but I mean, if you look at your pink triangle, you
yes,
have a hypotenuse on that triangle that's going up on a certain angle.
Yeah.
How do you figure out what that angle is?
So what we did is we didn't explicitly figure out that angle. So the thing is, what we did is we used the compliance and the inertance and elastance equations to be able to estimate the resonance frequency, because from Dr. Hentos's 2015 paper, he had an equation, a ratio, if you will, 1/2 pi times the square root of, I think.
Oh, I see.
inertance
So
and
you
elastance to be able to
fit
estimate.
that equation to the previous points.
Exactly.
Calculated whether, I see.
That's right.
Why would you even do that though? Because I think the Restmon device just truncates straight up. It doesn't do any integration beyond 19 hertz. If you wanted to compare how this methodology works with the Restmon Pro method, unless I'm wrong.
I have no, do you really, because I
I'm pretty
don't,
sure that's what they do.
because
Charlie, since you were involved in leads, can I pick on you for technical
No.
or is that the?
Yes, when I pulled up the Excel database, they show the other, but I don't know how AX is actually calculated. So that,
Their device can do the full spectrum, but my
Right.
understanding is, at least that if I remember it correctly, they used only the tri-frequency.
well, that was because it was an earlier model of the software. But today it's a little different.
yeah, the current software is that if actually, like, if you make a measurement and the resonance frequency is greater than 19,
Right.
it triggers an error, and then they ask you to actually redo the study
Got it.
with the full spectrum.
Spectrum, okay.
But so I don't know actually what their AX measurements are.
Okay.
So so here, what?
Not as fussed about it because we didn't find a lot of difference between AX and just exit 5.
You'll see.
It's pretty much.
Yeah, you'll see.
Anyway,
Just a final.
Did you look at your data with ignoring the pink triangle as well, or that's? to be
You
done?
would.
Yeah, sorry. Sorry.
Yes, that's blood.
But
Poor guy.
But so.
Yeah, let him-
then
I'll come
like,
back to
So the FRES is interpolated using the equation, but then that pink triangle is just a triangle, right? So it's just triangle between 19 and.
Yeah, I'll ask a question after.
Yeah.
Yassine, welcome to the club. We only do this for people we like.
Thank you. No pressure here.
You're doing well. You finished a sentence.
Now, there is a clarification here.
Yes.
N is referred, the 564, is that number of patients?
Number of patients, that's right. Sorry, it's not the frequencies.
Oh,
It's frequency.
well, that I didn't. Just wondered if, I had another thought it could be, but I assumed it was the patient.
Yeah, it was
And
564.
So these are patients, correct?
Correct, yeah.
And, they're all COPD patients, asthma.
There's some mix.
So go back to your first, your date. So this is a histogram of those 564 patients and what their FRES is.
Yeah.
So why don't we let him finish?
Yeah.
And then
I appreciate it. All right, so where was I? I think we already went past the resonance frequency, lost a little bit of track. But long story short, what we got was when we were trying to estimate the resonance frequency with only truncated data for patients above 20 hertz, it didn't work out so well. As for the area of reactance, it was
slightly more robust. There wasn't a significant difference between the measured AX from the tremor flow and the estimated AX, even with the triangle. And when we look deeper into it, the reason why is that we saw is that the patients under 20 hertz, they were virtually identical. There wasn't much of a difference at all, right? However, for the patients above 20 hertz, although there was a statistically significant difference, we can see that from the box plots that the differences aren't as large as what we saw earlier with the resonance frequencies, right? So that brings us, to like the third or the second objective was, could we find a alternative metric? I'm sure you guys are going to enjoy this one. So long story short, what it is a truncated AX. So because of the fact that we're looking only at between 5 and 19 hertz, we wanted to see, can we find a way of differentiating between normal and abnormal within 5 to 19 hertz, right? Without having to try to predict or extrapolate or interpolate. So what we did here is truncated 19 hertz, where if the patient has a resonance frequency over 19 hertz, as the case is on the left, what we would do is we'd only look at the area under the curve from 5 to 19 hertz. There would be no additional estimation, no additional pink triangles. However, here's the pink triangle, by the way. If we have a resonance frequency underneath 19 hertz, what we would do is we'd estimate two triangles: the area under the impedance at 0, right, which I'm dubbing here AX1, and then AX2, which is the area under the curve from the resonance frequency up to 19 hertz. And what we would do is we'd find the difference between these two triangles, and that would be our truncated AX for patients who have resonance frequency underneath 19 hertz. So how well did that do? So the first thing is, it was robust whether we used sparse metrics or the 5 to 19 full range of frequencies. They were virtually identical. We could see here from the box plots on the left, but there was no significant difference. Second thing to note is that for patients under and over 20 hertz, there was also some, I guess, some split, if you will, in the density of the data. So usually what we found was that patients who had resonance frequency under 20 hertz would tend to have a truncated AX between 0 to 10 or so. And patients that had a resonance frequency over 20 hertz, it was higher, up to around 20 AX, truncated AX, right? Next thing I want to talk about is how does it compare to the more common metrics that we have now. So on the left, what we could see is a truncated AX to our regular full AX. And for the most part, it's highly correlated, except if you look at the bottom, there is a slight deviation there. The reason why is because there is the cases where the second triangle that you're subtracting from the first would actually result in a negative result, right? What we found usually whenever I was looking more into the data is that usually happened at around the resonance frequency of 11.5, give or take. Meanwhile, for the rest of the patients, it was found to be relatively robust and highly correlated to the regular AX. As for the correlation between the truncated AX and the X5, what we found was a pretty strong negative correlation as well. So to reiterate, to give the takeaways, both the AX and resonance frequency, when we're trying to estimate them with a reduced range of frequencies, they obviously suffer, especially for patients who tend to have non-normal lung physiology. This proposed measure is robust at all ranges, and also is similar regardless of whether you use a sparse 3-point or the 5-point measures. Thank you very much. I'll be happy to take any.
Thank you very much. Are there questions?
Let's just say, I actually like this because it's using all the data, and it's giving you a single number that uses all the data. And I see no reason why it wouldn't be just as useful as AX. There's nothing magic about AX. But you don't have the problems you alluded to. So I kind of like it, actually.
Yeah, and it was a thought of mine as well, right? Where it would be great because, like you mentioned, especially what we mentioned yesterday, what Charlie mentioned, right? Where the clinicians usually, they just want a number to be able to say, is it good or is it bad? And as far as I've seen so far with truncated AX, it does help give you that number.
It looks more sensitive at low values too. Like it's got a dynamic range.
Yeah, that's right. Yeah.
And that's what you kind of find, but So I like it too, because, it means that you're not crunching in a whole lot of data in terms of using it celebrating. I was confused by, and when we do clinical trials, anything that's going to reduce the variance of whatever endpoint we choose increases the statistical power.
Correct.
More importantly, means we have to use fewer subjects or patients.
That's right.
So the one thing that I'm struggling with is these are patients. So I'm going to get back to what I got cut off at truncated head.
Right. So there was a mix based off her data set. It was predominantly patients who had either interstitial lung disease or they were lung transplant patients. There were also some normal healthy patients, and there was a third disease, but the name is too long and I don't remember at the top of my head.
Well, as we saw yesterday, there are situations where AX changes, but X5 doesn't, and that presumably is a change in the resonant frequency because it's the only way you're going to get an increased area.
Right.
And the other is that in patients with really severe airways disease like COPD, resonance can be really high, in excess of 60 hertz. So it's, so how this would then perform in those, probably not as important because what we're looking at is early detection of early disease.
Right.
And then once we get into more severe disease, an idiot can figure out that things aren't going too well when the numbers are super duper high.
Right.
We don't need to get into the subtleties. It's only when we're going to get detection of really subtle disease in a general population, as an example. And then I missed it when you did the second AX.
Yes, I could go back to that graph if you'd like me to.
Yeah, I'm having trouble about understanding.
Yeah, I wanted to bring
Why
that
would
up.
you?
And you're introducing another negative number that clinicians just love.
Oh, that'll.
I would just do the integration of tilde resonance.
But I don't know what it means.
Fenway wants to defend her position.
Can I
Defend my position.
I join the objection.
Yeah,
I
think.
Well, I'm willing to accept it's a good idea. I just want to understand what the rationale.
But can I give you two rationales? First of all, it makes it more sensitive, gives a bigger dynamic range. But the other thing is I would caution anybody about asking the question, what does it mean? That's a fool's errand. Don't even go there. Use a purely empirical, probably sensitive measure.
Okay, that I buy.
Correlate with something else.
Yeah, that I buy.
Okay.
I'll go with that.
Right. I can never, Jason is the final word here, but I also want to make this point. Lung function testing, whether it's oscillometry or spirometry, does not diagnosis mode. So it's a diagnostic test. It's completely agnostic to the disease. And so that, I take your point about the severe COPD patients, but I have in that cohort patients with very, very bad clot, chronic lung allograft dysfunction, and they look for all intents and purposes of really, really bad patients. So we have this full range of people. So I think like probably 120 of those are pure IPF patients with really bad interstitial fibrotic disease. And then we have this whole range of like the really lovely thing about lung transplant patients is their lung physiology is all over the map. And so we have in that specific population people who are many years post-transplant, and then a smattering of early post-transplant with normal restrictive.
Yeah. And actually,
Can I actually?
Okay.
I'll ask someone
Oh,
else.
yeah.
There was also another question.
Yeah, and that, or get to you, but Thomas, if you have a comment on this.
Yeah, so as a biomedical engineer and having trained with Jason, I like this very much as well. It's very mathematically proper and correct. As a manufacturer, I'm wondering, this is, AX is an established parameter. It's been the Goldman triangle. It goes back to, decades at this point. And it is well established in the literature. There's a big body of literature. There are lots of reference values. So I guess the question is, do we really want to propose this change? AX is gaining momentum and helping the adoption of oscillometry. So do we really want to rock the boat by changing one of our most appreciated parameters, invalidating our reference data, having to recalculate all that? And if so, do we want to call it AX, or do we want to call it perhaps something else so that at least it's distinguished?
Well, can I answer? I think it's a very good point. I definitely don't want to call it AX.
Yeah, sounds good.
I think my immediate reaction is keep pushing AX for adoption. And when it gets established, think about this type of thing and maybe introduce it. It's sort of related to that. You know, if you have data that goes above the resonant frequency, people tend to think of AX, I think, as being indicative of stiffness. But AX itself involves inertance. And the more you go above the resonant frequency, the more it's going to involve inertance. That's why don't even go there. Don't even think, what does it mean? In fact, if you fit the RLC, the resistance alertance elastance model, to the curve and calculate AX, there's an analytic formula for it. It looks god awful, and it involves E and I. You look at that, it makes no obvious sense at all. So it's not helpful to think what it means in terms of physics. It's purely empirical parameter. And I think if you have something that's robust and sensitive, a little bit like FEV1, you know, what the hell does that mean? But it's robust and sensitive.
It's robust and sensitive, but I think it's more heavily weighted by elastics than by inertance, and especially because of the shape. I don't agree, actually. The more you go this way, the more it's affected by inertance, because it's just narrower and narrower. And the more you go out this way is probably because of reactance or X bringing it down. And so it's actually more affected by like elastics, the more it goes out.
That may be the case.
Yeah, so I think, and it has unity of elastics. So we shouldn't call me a fool for trying to give it.
Sure, I'll call you.
But it sometimes tends to be less noise, but it's the one that's statistically proven for smaller end.
Well, sure, it uses all the data.
It uses more of the data.
Yeah.
And I think it probably in the long run will more represent elastance than any. I don't actually buy that it's terribly affected by inertance. I really don't buy that you want to subtract a more inertive triangle from the triangle, so that thing is I oppose. But I think AX is still open to interpretation. I would not say it's just a number. That's me. Okay.
Have you seen the formula? I'll show you.
You can.
And
We're gonna make
and you just fit a curve that will give you an estimate of FRES, and then you can calculate the AX. So just take 5. I think 5 points would be better to fit a curve. Fit a curve, linear curve. Say, okay, we either hit the moon or we passed it.
But I think that is what we currently do, is that we, like, when we look at a data, they're like, you see that like 40% of those patients, right, have FRES greater than 20. And so we have a whole bunch of people who, like, about 40% will never cross. And so I think I mean, you know, Thomas, you can, right? Because I think what we do now is if it doesn't cross, you assume that it's 37 and you calculate the AX that way. However, you know, I don't know if there are other device manufacturers here, but I've never been able to get a straight answer in terms of how those other measurements are calculated in the other devices. And so we've been playing around with the new Resmon, and it, you know, it's when it doesn't cross, you know, it's greater than 20, and then it's, well, you know, this test is enveloped, switch to a different frequency. But if the goal is to actually use this as a test to look for disease, you know, having something that, you know, you can sort of more accurately, so more sensitive in terms of, you know, using the frequency ranges between 5 to 20, I think is helpful. And the reason for subtracting, you know, that big triangle above is that it provides you with a more sort of sense of range because all those folks are going to be
Well, with, I think we have to move on. Sorry. We have to move on. I think this is where we are. We basically have introduced a new term and it may survive and not. But
We got
it's a good
more people to look at their data this way as well. I would encourage people with data sets to explore this.
So thank you very much again. Yeah, okay, you can help me with that, that's great. So we go online to Hungary. Sarah Nemeth, University of Szeged, Hungary. Are you there? Great. Perfect.
Yes, thank you for the introduction. I will share my screen. Okay. So, as it was mentioned, I will talk about intra-batch measurements and also data processing. After talking so much about intra-batch, I will give a little overview of it. So, conventional multi-frequency oscillometry focuses on the frequency dependence of impedance, and it reports average values for inspiration and expiration over several breathing cycles. Intra-batch oscillometry uses a single oscillation frequency, and it tracks down the changes in resistance and reactance for each breathing cycle with efficient temporal resolution. This oscillation frequency is set to 10 Hz for adults and older children. On the diagram, you can see the steps of data processing. As you can see in the first row, we use pressure and flow signals. To begin, we filter this data with a bandpass filter using the corner frequencies of 8 and 12 Hz to eliminate all unwanted signal components such as breathing frequency and its harmonics, as they distort the underlying respiratory dynamics. Next, we divide the field data into smaller segments of 0.1 second to easily obtain the 10 Hz component with Fourier transform. And we finally compute the impedance by the complex division of pressure and flow components. As you can see on panel A and B, there are the flow and pressure signals, and panel C represents the impedance that we calculated, as I explained before. However, we are working with impedance loops, as they give a better insight to the dynamic properties of the respiratory system, including the nonlinearities that arise due to the dependence on flow and volume. These impedance loops are visualized by plotting the impedance as a function of flow and volume, as seen in panels D and E, and I will call these volume and flow loops from now on. To illustrate this concept, we use the recordings from a subject with tidal expiratory flow limitation. So here you will see how the flow loops form as time passes. On the left-hand side, you can see this process for a healthy control patient. As you can see, the resistance increases with inspiratory and expiratory flow, and this reflects the nonlinearities of the upper airways. The reactance reflects its behavior. We focus on tidal expiratory flow limitation, which was first addressed with intra-batch oscillometry by Delaca and colleagues. It is an extremely severe mechanical abnormality, which can be detected by spectral oscillometry. It occurs when airways narrow excessively, reducing expiratory airflow. On the right-hand side, you can see this process for a patient diagnosed with COPD. Upon reaching peak inspiratory flow, reactance does not change much, nor does it during the first half of expiration, but after the peak expiratory flow, it deviates from its previous trajectory and it decreases dramatically, reaching its minimum at a low flow value that we will call limit flow. And at the end of the expiration, this flow limitation releases due to inspiratory activity. Generally, no significant changes in resistance are observed. Here you can see an example of COPD patients' measurements broken down into individual loops, as we did some data cleaning, and it helped to eliminate all irregular loops affected by artifacts. Using the measurement data of patients with known conditions, we trained and evaluated different machine learning models. These models give an output of a number which is between 0 and 1, and it indicates the probability of EFL presence. To achieve this, we cleaned the data and extracted features that could robustly describe the fine mechanics of the repository system. For example, the loop area, and we included some shape descriptors such as circularity, and we employ different feature selection and processing methods for training, and we maximize the information extracted from this data set using cross-validation techniques. The confusion matrix describes how well our model can differentiate between healthy individuals and individuals with flow limitation presence. As seen, our model can identify EFL with 97% accuracy on this data set. Additionally, we wanted to evaluate measurements of patients with obesity hyperventilation syndrome. This is a major cause of chronic respiratory failure, which affects individuals with obesity, and it worsens nighttime breathing. OHS arises from a complex interplay of factors such as excessive thoracic and abdominal fat, which compresses the lungs, and it restricts its ability to fully expand. And also upper airway issues such as narrowed passages, which can further impede airflow. Bagyash and co-worker measured 35 patients with obesity hyperventilation syndrome in supine position at progressively increasing levels of continuous positive airway pressure, and we analyzed their data. We evaluated these OHS measurements with our model, which gave a gradually lower probability to the presence of EFL as the CPAP level increased, which can be seen in the figure. The first row shows the volume loop, the second row shows the flow loops, and the third row shows the output of the machine learning algorithm for each CPAP level. The first column shows the measurement without any repository support, and this is followed by 5 centimeters of water increments in CPAP, where the EFL pattern gradually fades as the repository support keeps the lungs open. Different patients require different level of support to decrease EFL. In the last column, the patients were remeasured with 0 support, and as you can see, the baseline pattern returned. Now I would like to show some extreme cases, and by that I mean in the first figure, the patient flow limitation almost immediately disappears, and on the left side, the patient's EFL persists even despite 20 centimeters of water support. And this figure shows the output of the machine learning algorithm at a different level of repository support for all measurement data. It can be seen that the probability of EFL is high at 0 support.
We lost you if you can hear us.
Really? You cannot hear me?
Yeah,
You're back.
Loop back 30 seconds if you could.
From this slide?
Perfect, okay, go.
Yeah, if you want, turn off your video. That can help the communication sometimes.
I turned on my video.
No, turn it off now.
Yeah,
So
I see. Oh, thanks.
Because we're getting a signal that it's unstable,
I see.
or we're unstable.
Okay.
Yeah,
You're
perfect.
So here you can see the output of our model, and it's important to say that it gave lower probabilities to the EFL presence as the CPAP level increased, and in the first row, you can see volume loops. The second row shows flow loops, and the third row shows the output of the machine learning algorithm for each CPAP level. And the first column has 0 CPAP, and it is followed by 5 centimeter of water increasements in CPAP. And the last column, it is a remeasurement with 0 support. And it shows that the baseline pattern returned after the support stopped. Yes, so here are 2 extreme cases. The first one where the flow limitation disappears almost immediately, and the second one is where the pattern persists even despite 20 centimeter of water support. And this is the figure that shows the summary. It shows the output of the algorithm at different level of CPAP for all the measurement data. And you can see that the probability of EFL presence is really high at 0 support, both at the beginning and at the end of the measurements. And as the CPAP level increases, the pattern gradually fades for more and more patients at different levels. And also, I'd like to talk about a little bit about limit flows. For EFL patterns, the flow value associated with the most negative reactance is called the limit flow, as I mentioned in the video. This is also present in other EFL associated diseases. The estimated values of this limit flow in different disease groups are shown in the figure. The first column shows patient with COPD. The second column shows with patient with ILD who has flow limitation. And the third one is patients after lung transplant who also show flow limitation. Finally, the OHS patients we just studied with 0 CPAP. The limit flow for OHS patients is higher than for the other groups. This is because the lung closure in OHS is more uniform compared to the obstructive diseases where the closure can be heterogeneous. In conclusion, we use machine learning to help with quality control and identifying pattern. We found a significant presence of EFL in OHS patients in supine position, and this pattern that we observed was really similar to the one was observed in patients with COPD at different levels of CPAP. Thank you for the attention.
Very much. Wait, you turned your video back on. All right, do we have any questions? Perhaps I'll take the first one then. So excellent. I very much appreciated the talk, but I wonder if attributing it to the same mechanism as we see in COPD. So this is the expiratory flow limitation, because you can have different reasons for changing the stiffness during breathing. For example, the pressure volume curve. If you're moving up and down and the slope happens to be steep, I might have a different elastance at the end of inspiration than I do at the end of expiration. For example, ILD is a good example where we have very stiff lungs. And so are you sure that it's flow limitation or it's just a delta in reactant in the electrons at 10 Hertz in this case?
Well, for this, I'd like to refer to Zoltán Hantos, as I was mainly working on it as a computer scientist. I would like to ask him to answer this question.
Okay, I see Zoltán.
Oh, to make you
He's running away, I think.
So, Jeff, it's a good thing. Expiratory flow limitation can be at any generations of the airway tree, and upper airways can be included. But this peaky behavior reaching the limit flow of 0.1, 0.2 liter per sec minus. This is a very characteristic pattern which can't possibly be explained by any other mechanisms, but the peripheral airway closure, which is not manifested much in the total resistance because these are low resistance, these are the silent zone tubes. So first of all, you detach lots of compliant units without noticing any change in resistance. If it is associated with resistance, so this negative peak in reactance is associated with a positive peak, which is not uncommon, then it indicates that you are moving more centrally and involving the resistance generations, in fact. So I think this pattern is very characteristic and can't be, what to say, misinterpreted.
Yeah, well, I think I agree. And I think if it's of the pattern that you're ascribing to the smaller airways, the more peripheral, it wouldn't be, I don't think we would call it the usual, we wouldn't, when people think about flow limitation, they think about high expiratory flow limitation. But in this case, I think it's the most extreme flow limitation where the airway just is so closed. There's very little flow of the oscillations going through that airway, or none at all if it's actually completely closed. So flow went to zero. No, it's what we would call de-recruitment. Jason.
Yeah. And also, if you're dealing with COPD patients, you have a reasonably high pretest probability that that's what you're going to find. And so it seems like a reasonable patient. I'm wondering, are there any situations, maybe in asthmatic patients or people suspected that, where vocal cord dysfunction look like this or confuse the situation? In fact, what does vocal cord dysfunction look like in terms of your plots?
That's a very good question, Jason. Actually, which we badly need at the moment is a similar data set from asthmatic patients. We are very keen to have them, but at the same time, we acknowledge now the COPD material received from Ron's pulmonary practice. And likewise, the ILDs and LTX data from Chang Wai. So these were the basic, and first of all, the healthy controls, which are very, very, very difficult to access to for 10 Hertz intrabat studies. So this is a preliminary study when we had two populations, one without TFL and one with the tidal EFL. And these very obese hyperventilation syndrome patients were classified into one of these groups. Probably, if we have a more wider spectrum of major pulmonary diseases, which I expect for the last week of one coming month or months, that our spectrum would be enlarged and
let me translate for the audience. Goldie is taking me to task internationally for procrastinating on sending him data that I've yet to organize, and that I've been promising him now for, I think, 6 weeks. I'll blame it on my students that kept me very busy.
Oh, you're doing well. And then, that's so good.
That's right. It's a poor man that blames his tool.
Okay.
So
one more question. If you...
Yeah, it's more of just a general comment. I love this approach, and I think this is like really relevant to some of the problems that Nadia was talking about yesterday with interpretability of this like wide spectrum of data that we get from oscillometry and other clinical variables. And also, I've watched Kate overread so many data sets. And it makes me so sad how much time she spends doing it. And I would love to see this sort of approach applied to quality control as well, right? Because I think all of us have labeled sets of what we think are good measurements or not good measurements. And I think the machine learning approaches are the future of our field. So great work.
All right. Well, thank you very much, Sarah.
Thank you.
Okay, excellent segue, because the next talk is involving incidence of small airway dysfunction and one kind of follow-up study. Right. So, is it? Oh, it's virtual. Are we there?
Good afternoon, everyone.
Okay, could you share your screen, please? It'd be wonderful. Should go back to the.
Is it visible?
Yes, it is.
Okay, good afternoon, everyone. My name is Botan Sipos. I'm a fourth-year medical student at Semmelweis University, Hungary.
Yeah, could you do presentation mode, please? F5?
F5.
Or the little screen icon at the bottom?
It's not visible?
Oh, I think you're on the wrong screen. You're sharing your other screen then.
Oh, sorry. Maybe now?
That's it.
Yes.
Very good. Okay.
Okay, sorry.
Take it away. No, thank you.
So my name is Botan Sipos. I'm a medical student at Semmelweis University, Hungary. And I'm honored to talk to you about the detection of chronic lung allograft dysfunction using intra-breath oscillometry. We have some common things to say with Shari in the previous presentation, so I would like to apologize in advance for any repetition. Chronic lung allograft dysfunction is the most common cause of morbidity in lung transplant patients after the first year post-transplant. It's responsible in this period for 40% of all mortality. Its prevalence in the first 5 years post-op is 50%. In clinical practice, the diagnosis of CLAD is based on spirometry with supporting data from chest imaging and other clinical data. CLAD is defined as an at least 20% persistent drop of FEV1 forced expiratory volume in 1 second compared to to a baseline value, which is the mean of the two best FEV1 values measured post-op, taken at least 3 weeks apart. There are several manifestations of CLAD, the most common being bronchiolitis obliterans syndrome, which is obstructive in nature, but there is a restrictive phenotype too, which is restrictive allograft syndrome, that is much less common, but has a worse prognosis. Our retrospective study aimed to investigate the appearance of tidal expiratory flow limitation during spontaneous breathing in lung transplant patients using intra-breath oscillometry. Our hypothesis was that the appearance of expiratory flow limitation was correlating well with the appearance of CLAD. The ability to detect expiratory flow limitation with oscillometry was first described by Delapez Research Group in 2004, based on the fact that during expiration, in case of EFL, the reactance falls significantly. The mechanism behind this is that there are choke points, flow limiting segments in the airways in EFL, through which the oscillatory signal cannot pass to reach the alveoli, which are contributing the most to the lung compliance. So it makes a lowering in the lung compliance and thus a fall in airway reactance. The Toronto General Hospital lung transplantation clinic has been using ThermoFlow devices with 10 Hertz intrabreath research modalities in routine follow-up of lung transplant patients since 2018. In this research, we analyzed the intrabreath measurements taken from July 2020 to...
Botan, you're breaking up. Can you maybe turn off your...
Sorry?
Your audio is breaking up. If you maybe turn off your screen, that might help. Thank you. Good. Your camera.
Is it good now?
Yes.
Do you hear me now?
It's off and on.
Okay, so we have been sorry, maybe my connection has some problems. Do you hear me now?
Yes, pretty good.
Okay, sorry. We have been analyzing the oscillometry data from a cohort of patients who underwent lung transplant surgeries before 2017. This group consisted of 171 patients with altogether 708 measurement sessions, each sessions including 3 consecutive measurements per patient. We evaluated the incoming data using a software developed by our research group. Every breathing cycle has been visualized as loop curves in graphs depicted airway resistance and reactance as functions of respiratory volume and flow. In cases without any restrictive or obstructive disorder, the dependence of resistance and reactance on volume and flow is minimal. The loop curves showed us different characteristic patterns, from which I would like to highlight the pattern of expiratory flow limitation. In this pattern, the reactance drops significantly during the second part of expiration, which appears simultaneously with a small lowering of resistance. This makes the resistance-reactance curve resemble a mirrored C-shape. This is important for us because it makes us able to differentiate the expiratory flow limitation, which appears in the bronchiolar, so the small airway level, from the pattern typical to upper airway obstruction, for example, the bronchomalacia of the anastomosis, where the minimal reactance can be measured at the same time when the resistance is at maximum. So the resistance reactance curve is kind of linear. From the 171 patients examined, 66 were diagnosed with CLAD based on spirometry. Most of them were BOS phenotype with insufficient data from patients who have restrictive allograft syndrome. We observed the expiratory flow limitation pattern in every measurements of 27 patients and in some measurements of 13 patients. So altogether, EFL pattern was detected in 40 cases. The patients who showed the EFL pattern continuously, 85% of them were diagnosed with CLAD before. In an attempt to quantify our results, we identified 3 markers which characterizes the EFL pattern while excluding the pattern appearing in upper airway obstruction. These patterns are the following. DXE, which is the decrease in reactance during expiration, X-min E, which is the minimal reactance during expiration, and AXV dot, which is the area enclosed by the flow reactance curve. using Mann-Whitney-Wilcoxon analysis to differentiate between clad and no clad groups using these parameters. The p-value is under 0.001 for all three markers. So the difference of these parameters in clad and no clad groups are significant. We calculated specificity and sensitivity of clad detection using these parameters, from which the results are shown here. Combining a designated threshold value of AXV dot and XMIN E, we get a very high level of specificity with a rather moderate sensitivity. Focusing solely on XMIN E, we get a balanced high level of both specificity and sensitivity. In conclusion, we can say that the TEFL oscillometric pattern was way more prevalent in those diagnosed with CLAD and rarely appeared in patients who hasn't been diagnosed with CLAD. These retrospective studies in line with the ANFU and research groups 2022 findings about the oscillometric distinguishability of CLAD and suggests the potential to use intra-breath oscillometry to study or even to predict CLAD. The limitation of our study is that these patients underwent surgery before the introduction of intra-breath oscillometry to routine follow-up. So in most cases, also the spirometric diagnosis of CLAD predates our first measurements. Therefore, we are currently examining a data set of nearly 600 patients and 7,000 measurements, where the oscillometric follow-up started immediately after surgery. Our preliminary results also seem to confirm the hypothesis that the presence of expiratory flow limitation pattern is strongly connected to the appearance of CLAD. Thank you very much for your attention, and I would like to thank my supervisor, Professor Zoltán Hantos, and the colleagues of Toronto Lung Transplant Clinic, Dr. Changwai Cho, and Fu and Joyce Fu for all the help provided. Thank you very much.
Thank you very much, Gautam. That was excellent. Okay, other questions?
Well done. Really nice.
Thank you.
I guess I can ask one. So it's a similar question that I asked Sarah. In what would be the mechanism for the flow limitation in lung transplant patients? So what we have is a disease with obliterating the small airways. Are we measuring that directly? It's the obliteration of the small airways.
So yes, bronchiolitis obliterans syndrome makes narrowing in the small airways, and this makes the EFL pattern shown in oscillometric data, and that's why the resistance for which the small airways doesn't contribute that much makes a little fall. So in cases of upper airway obstruction, also the resistance rises, and that makes us able to differentiate the pattern of small airway disease and an obstruction appearing in the upper airways.
Yeah, so why is it more pronounced on expiration than inspiration? Why are we getting that narrowing of the airways or on expiration in this disease? It's not a loss of tethering like we see in COPD. It's something else.
If I'm not, yep, sir.
Charlie might have the answer.
Well, these lungs are denervated, right? Yeah, so they have no neural tone, and you're oscillating them at 10 hertz, correct?
Yes, it's 10 hertz. Sorry, just the connection is
Yeah.
very bad and it's
Right.
hardly.
So I would speculate that when you inhale, it's sort of like CPAP. The airways are held open, stented open, if you want to think that way. But on expiration, because there's no neural tone, they're starting to narrow. You see that on
Yeah,
low volume
CT,
too?
well, when we do inspiratory, expiratory CT, you see on the expiration, areas that are blocker and areas that are, in other words, some areas are
Yeah.
emptying and some areas are not empty. That you clearly see on CT on the inspiratory.
Okay, so that would argue against nerves because that would be ubiquitous. Whereas this is more here and there.
Patchy, yeah.
Well, another thing too is, I mean,
But it's still dynamic,
Yes,
right?
no, absolutely. You won't see it on the inspiratory scan, but on the expiratory scan, that's where you see.
But on flow volume loops, do you airflow limitation as well?
Do, and particularly the patients with the boss phenotype, they actually really look like emphysema.
Got it.
More so than like chronic bronchitis, they look, the look like. And the inflammation is really more like it's peribronchiolar.
Still.
the inflammation presumably is critical. Your immune system's attacking them. So you have an inflammatory process going on. So why wouldn't it be similar to what happens in asthma? The narrow, thick airway, narrowing of the lumen, pronounced flow limitation, for all the reasons that.
Yeah, but asthma
we
is
have
both
some-
inspiratory and expiratory.
Well,
Whereas this one,
mild asthma.
yeah,
That?
it's asthma is more inspiratory or it's both, right? But COPD, the narrowing is expiratory.
Expiratory.
But in bronchiolitis, it's expiratory. Bronchiolitis obliterans. So there's something wrong with the peribronchial space that's weakening the
Right.
parenchyma.
But
And it's,
I
so it's
mean,
narrowing
in
during
asthma, it's
expiration.
still mostly expiratory flow limitation.
Not, well, the loops in asthma are not as much like this. They're different. They're
Only it's
well,
severe.
Zoltan knows the difference better than I
If
do.
it's severe, then yes.
In severe, yes, yeah.
Yeah.
But in
Sure.
mild asthma, no.
Yeah.
All right, well, all right.
But when you look at dynamic CT images in these folks, is there narrowing on expiration?
There is some emerging, and so what you see is that the airway thickness increases, and then what we heard yesterday about the pi, which that is also increased, but it's very early data, and the problem is that some of, for example, like the cohort that Button is studying predated 2017, so we don't. What's kind of unfortunate is we didn't have a protocol to have inspiration, expiration CTs. We had CTs at all time points, but the protocols for the acquisition is different.
Good job.
Are there no more questions? So well, that brings us to almost the end. Thank you very much.
Thank you very much.
And also thank all those in Sega, Sarah as well, and Gold.
Okay, well, thank you for moderating, Jeff, and well, indeed, so I will close down the seminar with a thank you to all of the participants, the speakers, the industry partners that made it possible, and my students for the help and the logistics, and just to part with the idea that I would never throw my students under the bus. Solely, I've had to make priorities in terms of data, presentation, information, and I have chosen to mentor my students above sending you data. So there you go. So thank you.
Witnesses.
So thank you very much, everybody, and I hope we can all do it again next year. I don't know about you, I find this very helpful, and I hope that the dinner will be on your agenda for those of you who are online and you'll actually make the trip up to Montreal. So have a great rest of the year, everybody. See you in Vienna or in San Francisco or somewhere in between over the next year. Thank you very much.
Thank you, Ron.